All righty. Any questions before we get started this morning? All right. Stunned silence. Always enjoy that. Let's talk game plan then. Our game plan for today, Tuesday, July 7th, is going to be what it's going to be for the next week, basically. In lecture, we are going to be doing muscle physiology. And then in lab, we're going to be focusing on our origins, our insertions, and our actions. So you're going to see that for our lecture and lab game plan, basically for the rest of this section. Again, it feels weird not having Thursday on the schedule. Again, I want to make sure you guys know to come on Thursday. So we do have class on Thursday. There's just nothing due. We do have 4 assignments due still left. Your 2 unit reviews, 10 and 11, your Physio X exercise 2, which has 7 activities to it, and then that 20-point muscle activity that is going to be graded for correctness. And again, just like before with the last review, I encourage you to work together in groups on these to come up with them, and that is totally fine and acceptable. Just make sure you're writing the answers in your own words. And it really goes hand in hand with what you guys are planning on doing for your group presentations that will be starting today as well. And again, we're just going to rattle through those quickly as we do that. And again, it's just a way to add some light and levity to what is a very dense topic. All right, and that's it. That's our game plan. Pretty simple and straightforward. So if there aren't any questions, let's go ahead and get started. We left off last time, and as I mentioned, we were getting ready to talk about our big physiological process that is that muscle contraction. Again, I want to emphasize this is a continuous process from the point where we make a decision all the way to the point where the muscle contracts and moves the body through space. However, as I also said, we are going to take this incredibly long physiological process and divide it into 3 more easily digestible pieces. So for each of these pieces, obviously the 3 steps, we have to identify the step by name. One of the important things to talk about is we'll also talk about and identify the goal of each of these steps as well as we go through it. What is it that we hope to accomplish with that particular part of the step so that we can think about these things, and it'll help us to understand and divide it up and be specific in the activity. So let's talk about step 1 first. Oops, hold on. Clear that. Step 1 in our process is communication at the neuromuscular junction. I know we haven't talked about what the neuromuscular junction is, but we will. This neuromuscular junction is the specific location where our nervous system communicates with the muscle cell. And we will talk about the anatomy of that and describe that in detail. And so again, we'll talk about that. Because again, the important thing to remember about the contraction of skeletal muscle, it is voluntary. It starts with the conscious decision to make that contraction. So we have to then convert this information from the nervous system to the muscular system. We have to irritate the muscle cell. Remember, we said the muscle cell was irritated. It was going to produce a big electrical signal. We called an action potential. We know from when we talked about it before that our nervous system communicates with big positive signals. And those big positive electrical signals are also action potentials. So when I talked about us having a goal, the goal of step 1 of this process is, as it's going to say here, is to convert our neural action potential into a muscle action potential. This is a very special process, so it needs a very special location where that's going to occur. And of course, that very special location where it's going to occur is the neuromuscular junction. So that is step 1 of our process, communicating at the neuromuscular junction. Step 2 in this process is what is called excitation contraction coupling. Right? What does it mean to couple?
To pair up.
Yeah, to pair up, exactly, to bring 2 things together. And that's what we're doing right here. Remember, we started with the muscle action potential, because again, remember where we finish step 1 is where we begin step 2. So we start with a muscle action potential, which is great, but what we want the muscle to do is to contract, to start to generate force. So we start with the muscle action potential, but we want to generate force. And of course, it is going to generate force by contracting. Right. Our goal then is to get the myosin and the actin to interact, to generate the force so that we can move our body through space. And as we talked about last time, myosin always wants to grab onto the actin. Myosin always wants to be generating a force. So remind me again why our muscles aren't in a constant state of contraction?
Because of tropomyosin.
Yep, because of the tropomyosin and the troponin, those regulatory proteins, absolutely. And so when we think of the goal of excitation contraction coupling, you're absolutely right. Our goal is going to be to move the regulatory proteins so that the myosin and the actin can interact. And remind me again, what did we say we were going to use to move those regulatory proteins?
Troponin.
Remember, contracting is a wonky process.
Calcium.
So, yeah, there you go. We're going to use calcium to move the regulatory proteins so that the myosin and the actin can interact. That is going to be our goal of the excitation contraction coupling. Our goal is going to be to bring these two things together, right? We got to get that calcium out of the sarcoplasmic reticulum and move those regulatory proteins so that the myosin and the actin can interact. We're going to couple, bring together the excitation to the point where the muscle is able to contract. All right, so that is step 2 in our process. Step 3 in our process is what is known as the contractile cycle, or what is also known as the sliding filament theory. You may use either of those terms when describing it. I like contractile cycle because, of course, our goal is to produce the contraction, and cycle tells us something. What does cycle mean?
It's repetitive.
Yeah, it's a continuous repetitive process, absolutely. And that's what we're going to see with the contractile cycle. It is a continuous repetitive process. However, as we kind of hinted at, to change the shape of the muscle, we have to change the shape of the myofibril. To change the shape of the myofibril, we have to change the shape of the sarcomere. And as we already kind of hinted at, the way that occurs is the myosin and the actin are going to interact and overlap with each other. They're going to slide across each other's surfaces. And so that is also why this is sometimes referred to as the sliding filament theory. And of course, as I mentioned, our goal here is to, ultimate goal is to move the body through space, change the shape of the muscle, right? So, but again, this is we are going to generate our tension, right? Or again, like I said, produce a force, tension, either of those are good terms to use. That is going to change the shape of the sarcomere, which changes the shape of the myofibril, which of course, as I said, is going to change the shape of the muscle cell, right? And that, of course, is our ultimate goal, to produce movement. Right? So that's what we have. We have this contractile cycle, we have the sliding filament theory, where we're going to generate the force, where we produce the change, where we change the shape, and ultimately are able to make the contraction, move the body through space. All right, and here, hold on. Grab that, move it out of the way. Basically saying the exact same thing. All right. Questions on that? Now, obviously, we're going to go into these in much more detail, explaining these processes. However, if there was a fourth process, what would that fourth process be? Exactly. Once we contract a muscle, we don't want that muscle to be contracting for forever. So if there were a fourth action, that fourth action would be, to relax the muscle. Now, we can think of it as a fourth step, or the other thing we're going to do with each step, we are going to describe the process of initiating the step and terminating. So basically, for each of the steps in the process, we are going to first describe how we turn it on or start it. And then we're also going to talk about how we turn it off or stop it. So with the muscle relaxation, we can think of it as two ways. We can think of it as a fourth step that occurs at the end, and we will actually talk about and describe it that way. But we can also, for each of the three individual steps, talk about how we initiate that process and how we terminate the process. And those would be the keywords I would use on the exam. Describe how you initiate contractile cycle or terminate contractile cycle. Those would be the types of essay questions you could expect. In fact, if you remember correctly, where do I have space? I'm going to cheat and do this here. I'm just going to steal a little of the board here. If you remember correctly, I mentioned that I guaranteed you there would be at least 2 essay questions on these 3 processes, or 3 steps, let's say it that way. All right? You guys hopefully remember I said that. However, it is a little bit more tricky than that, because if you think about the type of possible essay questions now, we could have the process of initiation, and I'm just going to abbreviate it, step 1. But I could also have terminate, step 1. I could have initiate, step 2. I could have terminate, step 2. I could have initiate, step 3, and I could have terminate, step 3, right? So all of those are possible essay questions that could be asked on the exam. And notice there's also a couple gaps in my list as well, because if we truly understand a process, one of the things that we can actually do to that process is modify it. And when we modify that step, we can see how it changes the muscle contraction process. I agree. And then just to round it off to a nice even 10, muscle relaxation as an entire process is something that could be. So there are 10 possible essay questions, and I guarantee at least 2 of them are going to be on the exam. So we need to go through all of these and describe all of these and make sure we understand all of these so that you can give me the detail and the content on this to be successful in the exam. All right? Sound like fun? I agree, it does. Sounds like tremendous amount of fun. Excellent. All right, perfect. Then with that in our pocket, let's move forward and start talking about step 1. Again, remember we are now on step 1. Step 1 is communication at the neuromuscular junction. Excellent. Spread that out. And remember, anytime we talk about one of our steps, it's always good to identify our goal. Remember, our goal here is to convert a neural action potential, which after we start writing this out as our essay question, we can then abbreviate as NAP into a muscle action potential, which we can then start abbreviating MAP. So we want to convert a NAP into a MAP. All right, that is step one. That is our goal. However, as I mentioned, we do need to talk a little bit and explain a little bit about what this neuromuscular junction is. So I've written this here. I don't think it's going to fit, but let's see if I can sneak it in. There we go, that works. So again, step one is communication between at the neuromuscular junction. And again, we basically have to communicate a signal from our nervous system to our muscle cell, right? We have to produce that muscle action potential or convert that neural action potential into a muscle action potential. We need to talk a little nervous anatomy here to make some sense of it. We've talked about what a skeletal muscle cell looks like. It is a big, long, cylinder-shaped cell like the one we see here, that we see in the picture. However, what we haven't talked about is, oops, no, didn't want to do that. What we haven't talked about is what a neuron looks like. Our nervous tissue is comprised of neurons. Obviously, we will talk the anatomy of neurons in much more depth when we get to them, but let's do some basic anatomy. Like most cells, it has a big, large, centrally located, let's change the color just to emphasize this, big, large, centrally located cell body. One of the things that makes neurons unique, though, is like we said, they're specialized for communication. Their job is to receive information and also send information. The way they receive information is they have these short, elaborately branched processes, right? Process is a word we learned back in the bones. A process is something that sticks out of something else. So there are these small, elaborately branched processes where the neuron receives information, and they're called dendrites. These are where it receives information. Then a neuron has one big, long, unbranching process that is known as the axon. And the axon is how it sends information. You have neurons whose cell bodies are located in the small of your back, and they go all the way down to your big toe. So if you think about it, that's almost a meter in length. Some of these axons are incredibly long, and they're completely unbranched, big, long superhighways for information to get to their destination. At the end of their destinations, what happens is the axon finally branches. And when it branches, it forms a structure we call an axon terminal. And at the end of that axon terminal is a specialized structure where our neuron is going to communicate with our muscle cell. This structure, again, as we've learned anything, anatomists love to name stuff. And as big as biology is, neuroscience is one of the huge parts of it. There are lots of different levels that this is studied. There's probably 10 plus different names that this thing is called. For our purposes, we will call it the synaptic end bulb. And the synaptic end bulb is this enlargement that we see here, where it is going to communicate with our muscle cell. So basically, this region right here, where our neuron communicates with our muscle cell, that area is what we call the neuromuscular junction. Excellent. Now, this neuron, of course, remember we talked about our skeletal muscle is controlled by a somatic motor neuron. But we also said that our smooth muscle and cardiac muscle is controlled by autonomic motor neurons. And neurons communicate to everything else as well. They communicate with lots of different things. So the neuromuscular junction is a specific term for a specific specialized area of communication. What we need is a generic term for where a neuron communicates with its target cell. And does anybody know the generic term we use for where a neuron communicates with its target cell? Bingo. It is called a synapse. So the neuromuscular junction is a specific type of synapse. All right? So this is a little bit of anatomy. Most of this we don't need to worry about. What we need to be focusing on is what's in the blue box. But we need a little bit of information to help us to understand that. So let's go ahead and do this. Perfect. And then I can do that. And then I can do that. Excellent. So the general term, again, is a synapse. Region of communication between a neuron and its target cell. So the neuromuscular junction, and I'm just going to abbreviate it up here, then specifically is the synapse between a somatic motor neuron and skeletal muscle cell. Okay? Does that make sense? Excellent. Complete stunned silence means yes, that makes perfect sense. Please continue.
Quick question.
Yes.
You said that's the specific, the one you just typed right now?
Yes, the neuromuscular junction would be the example of the specific. So on the lab exam, for instance, I could have a big bold arrow pointing right to this region right here, and I could ask you two questions. I could ask you to identify the general term for this region of communication, and your answer to that would be? Anyone?
Synapse.
Synapse. Or I could have this exact same one with this exact same arrow, and I could ask you to identify the specific region of communication, and your answer would be?
The neuromuscular junction.
Perfect. There you go. See, just that easy. Excellent. So let's describe the components of a synapse. Here we see the pretty picture that goes along with this, up close and personal. And for this synapse, and again, we are going to focus specifically on the neuromuscular junction because that's what we're looking at here.
Quick question. You said that the neuromuscular junction is a specific type of synapse between a somatic motor neuron and what? Skeletal muscle. Thank you.
Skeletal muscle cell. Excellent. So when we talk about the neuromuscular junction, or any synapse, but this synapse in particular, it has three components. And that's what we see in this picture. Component one is the synaptic end bulb. The synaptic end bulb is the specialized neural component. Right? As we talked about, it is the specialized end of the neuron. And I keep saying it's specialized. So we need to describe how it is specialized. The first way it's specialized is by how it's going to communicate. Notice, as we talked about, that action potential travels down that axon, a full meter from your small of your back down to your big toe. But when it comes time to communicate with the muscle cell, it doesn't actually give an electrical signal to the muscle cell. It stimulates it chemically. So our synaptic end bulb stimulates the muscle cell chemically. It does that by releasing a chemical signal. All right? So we need a generic term for the chemical signal. Anyone know what the generic term for the chemical signal that a neuron releases to communicate with its target cell? Like I said, we haven't talked nervous system yet, but it's not excellent. It is indeed a neurotransmitter. Excellent. There have been over 100 neurotransmitters that have been identified in the human body. All right, oops, I spelled it wrong. Transmitter, there you go. That have been identified in the human body. Here, at the neuromuscular junction, there is one and only one neurotransmitter that is used. So here, at the neuromuscular junction, the specific, and I'm going to just abbreviate it, neurotransmitter, is acetylcholine. Oops. Now, the good news is, once you've correctly spelled out acetylcholine once, you can then abbreviate it ACH. So notice if we look at our synaptic M bulb, one of the specializations of it is it contains many vesicles. Well, let's say it this way, has many vesicles that contain acetylcholine. That way it can release that chemical signal to communicate with our muscle cell. Now, releasing that acetylcholine requires a tremendous amount of energy. So notice there are those red squiggly things in there. So one of the other specializations of the synaptic M bulb is it has lots of mitochondria. So that it can release that chemical signal. However, do we want this chemical signal being released all the time? If it was being released all the time, it'd be telling the muscle to contract all the time. And do we want our muscle contracting all the time?
No.
No. So we want to have a way to control how it releases its neurotransmitter. And releasing a neurotransmitter is a pretty wonky thing. And what tells cells to do wonky things?
Calcium.
Calcium. So the third thing that our synaptic M bulb has are voltage-gated calcium channels. Now, this requires a little bit of a definition. All right? We've talked about channels before. Channels, of course, allow ions or things like that into or out of the cell. This is a calcium channel. So guess what passes through this channel? Calcium, excellent. And from what we know of the concentration gradients, which way is calcium going to want to go? Is it going to want to go into the cell, or is it going to want to go out of the cell?
Into the cell.
Into the cell, excellent. So we know calcium is going to want to go into the cell. But notice this channel is a gated channel. Gated means that it can be open or closed, right? Again, if you have, I can't think of a good example. When I was younger and when I would get in trouble, one of the things that my stepdad would do is he would take the door off of my door frame. So when I got in trouble, one of the ways that I got punished is I didn't get any privacy. If you take the door off the door frame, right, that's open all the time. Whereas if you have the door there, it can be opened, it can be closed, right? So with the door there, it can be gated, it can be opened and closed. And in particular, not only can it be opened and closed, but there is a lock that, or really a key that is going to unlock it. In this case, this particular channel is voltage gated. So in this case, the key that opens the door is a change in the membrane potential. All right. Now, remind me again, what is the resting membrane potential of a cell?
70. I can't remember what the other word is.
Millivolts. But is it positive 70 millivolts?
It's negative. Negative 70.
Negative 70. Negative 70 millivolts. Excellent. Absolutely. So in this case, with a voltage gated channel, when the cell gets more positive, it will open this channel. Now, do you think taking the cell from negative 70 to negative 69 is a big enough change to get it to open those gates?
No.
Probably not, or 70 or 68 or 67. No, a small change like that isn't going to be enough. But there is going to be a critical point. That critical point where a voltage channel opens, we give a special name to. And the special name to that is threshold. Threshold is the voltage a cell has to reach to open the, now just abbreviate it here, voltage gated channel. Oops, if I spell open right. Oops, if I still spell open right. There we go. To open the, and I'll go ahead and write it out. Voltage gated channel. So that is the threshold. And for this particular cell, in fact, for most cells, threshold is negative 60 millivolts. So notice it's basically a plus 10 millivolt change. That's all we need to do to the cell. We need to make the cell a little bit more positive, make it plus 10 millivolt change, get it to set negative 60. And if we get it to negative 60, that voltage gated calcium channel is going to open. All right, does that make sense? It's done in silence, excellent, that must mean it makes sense, perfect. All right, that is component 1 of our neuromuscular junction. Component 2 is what we call the motor endplate.
Quick question, sorry.
Yep.
Now, we said that if he has a plus 10, is going to open the channel. What happens
Plus
if
10 change. It doesn't have to get to positive 10 millivolts. It just has to get 10 millivolts more positive to get to negative 60.
Got you. Can you get more negative, or can you have a positive change of like 30? Does that make a difference? Or if it's open, it's open?
Right, exactly. If it's open, it's open. So if you give it a plus 10 millivolt, I mean a plus 20 millivolt change, it's still, I mean, once you open a door, you can't open it a second time, right? So, or open it more. So yeah, no, once it's just, think of it this way. I think of it in terms of a light switch. You have a light switch and your light is off. And if you just touch the light switch, nothing happens. If you put a little bit of pressure, nothing happens. A little bit more, nothing happens. But if you push it hard enough, the light switch goes up and the light in your room goes on, right? That makes sense?
Yeah.
Okay, if you push that light switch twice as hard, does the light come on twice as bright?
No.
No, once you flip that switch, it's on. So it's got 2 settings, open, closed, right? So once you open it, it's open.
Okay, gotcha.
Closed, it's closed. So it doesn't, if you can go more than that, but you don't have to. As long as you reach that critical point, and that's what threshold is, that critical point you have to reach. As long as you reach that critical point, then it opens the door.
Okay.
Great question. Any others? All right, let's talk about our second component of the synapse, and that second component is the motor endplate. This is the specialization of the muscle cell. Now, we keep drawing our muscle cell as this big, long, cylinder-shaped structure. I'm going to draw this, but then I'm going to erase it in just a moment. As this big, long cylinder thing. However, where our synaptic end bulb communicates with it, there is a specialization to the plasma membrane, a specialization to the sarcolemma. So it's a specialization to the muscle cell, but really, it is a specialization of the sarcolemma. One of the specializations to the sarcolemma is, as you can see from the illustration, it has many invaginations. What's the advantage of having those many invaginations, all those nooks and crannies? Well, if I have two cells communicating with each other and they have flat surfaces against each other, or I have two communicating with each other with lots of folds and invaginations, we have a massive increase in surface area. Excellent. This increases the surface area for communication. Notice the other advantage, right? Why is that English muffin so good? Because it has all those nooks and crannies that can capture the butter in it. Well, remember, our synaptic end bulb is releasing a chemical signal. And notice that chemical signal can get stuck in those nooks and crannies. And if it stays in that nooks and crannies for a longer period of time, it can have more of an effect on the muscle cell. So it increases the surface area for communication, and it increases the time that that it can interact with the neurotransmitter. So that increased surface area for communication is very, very important. So that is one of our specializations. Be consistent. The second is obviously if our neuron is releasing a chemical signal, that chemical signal has to affect our muscle cell. And so you also have many chemically gated, or what your book also sometimes refers to as ligand gated, cation channels. All right, more fun with vocabulary. Let's think about this. Right, just like we did before. Here, I'm gonna get rid of my picture here for a second.
Can I ask a quick question about the point you made before? You said that it has more surface area and then it captures more what?
Of the neurotransmitter, the chemical signal that the synaptic end bulb is releasing. Okay, so let's think about what we were just talking about before. We now have a chemically gated cation channel. So first, it is a cation channel. So what passes through the channel?
Positive charge, is it?
Positively charged ions, absolutely. Because it's a cation channel, it is positive ions. And in particular, what are the two most common positive ions we find inside of the human body? What are the two most common cations that we've talked about in this class?
Sodium.
Sodium and potassium, excellent. Sodium and potassium, excellent. So that is what passes through the channel. However, notice it is chemically gated. So of course that means it can be opened and closed. And in this case, what is the key that unlocks the door?
Plus 10 or more.
Well, remember, that was when it was voltage gated. This one's chemically gated.
Oh.
So what chemical do you think it is that's going to do it? Acetylcholine, excellent. Oops, yeah. Acetylcholine, right, is the key. So acetylcholine is the key that unlocks the door, then sodium, potassium pass through it, right? Again, it's chemically gated. It is unlocked by a chemical. Your book, that animation we watched uses the term ligand gated, but ligand just means a molecule, right? So it's the same thing. Ligand, chemical, they basically just mean the same thing. So you can use either term that you like, but in this case, acetylcholine is what unlocks the door, and then sodium and potassium are going to be what flows. And remind me again, which way is sodium going to go through this cation channel? Into the cell or out of the cell? Sodium goes into the cell, and which way is potassium going to move? Out of the cell. Excellent. Perfect. So there you go. So those are two of our three components. But if you notice, there is a third component to our synapse. If you look closely at the picture, you will see that while we have this specialized structure, the synaptic end bulb, where we have this specialized structure, the motor end plate, the two do not touch. All right, these two do not touch. Instead, there is a space in between. It's a small space, but it is an important space in between. And that space is called the synaptic cleft. Synaptic cleft is the space between the motor end plate and the synaptic end bulb. And since I wrote them out before, I can abbreviate them there. Right? It is the space in between. They do not touch. Two cells do not touch. Excellent. Now, our motor end plate had some specializations to it. Our synaptic end bulb had some specializations to it. There's going to be some important stuff about our synaptic cleft as well. The synaptic cleft, of course, is outside of both cells, so it contains interstitial fluid. This is important because, as we know, interstitial fluid has a high amount of sodium and a low amount of potassium and a high amount of calcium, all those ions that we want to go through the channels that we want to go through. But there is also something else important here between them. Here in the synaptic cleft is a special enzyme. And that special enzyme is a mouthful to spell. Acetylcholinesterase. The good news is, on a lecture exam, once you've written it out once, you can abbreviate it, ACHE. And with a name like acetylcholinesterase, it is an ACE, so it's an enzyme. What do you think acetylcholinesterase does? What do you think acetylcholinesterase breaks down?
Acetylcholine.
Acetylcholine, there you go. It breaks down acetylcholine. There you go. So those are the three components of the neuromuscular junction: our synaptic end bulb, our motor end plate, and our synaptic cleft. All right, questions on that? All right, let's do two things then. First, I'm going to go ahead and clear all of these words. So we can just do it this way. Again, we have the synaptic end bulb, we have the motor end plate, and the synaptic cleft, and we've talked about those specializations. Again, one of the keys to our neuromuscular junction is that the neuron and the muscle cell do not connect. So instead of just passing an electrical signal back and forth between the two, right, instead it has to use a neurotransmitter, a chemical signal to communicate. Again, the generic term for that chemical signal is a neurotransmitter. The specific neurotransmitter used at the neuromuscular junction is acetylcholine. And so our synaptic end bulb stores and releases acetylcholine. Our motor end plate has acetylcholine receptors, those chemically gated cation channels. And again, there's the space in between. All right, this right here is what we need to do next. We need to talk about the process of communication at the neuromuscular junction. Now, to do this properly, like we've done in the past, I think it is going to be important to go through this at least twice. To make sure we understand it, we want to start first by talking about the anatomy. Then we are going to go through the entire process of initiating this communication at the neuromuscular junction. Once we're pseudo-comfortable with that, we will then terminate communication at the neuromuscular junction. And once we are comfortable with that, we will then modify communication at the neuromuscular junction. So that's going to be a lot of work. So to do that, we're going to do it in steps. And the first step in this process is going to be to make sure we fully understand the anatomy. I did it with all the pretty words. Let's do it with some, oh, not so pretty pictures. So I'm going to go ahead and clear our words from before. And what I want to start with right now is to look at the anatomy of this. So to look at the anatomy of this, what we're going to do first is we are going to draw our skeletal muscle cell. So here, hold on, I need some space to make sure this all fits. Perfect. All right, can you guys see all of that?
Yeah.
Okay, perfect. All right, so let's move this here. That's my skeletal muscle cell, and I'm going to cheat. Oh, hold on. Wait, sorry. Got to undo that anyway. Ugh, never mind. Got to make this black. Excellent. All right. So again, this is our skeletal muscle cell. Oh, that's going to be way too big. That's our skeletal muscle cell. Excellent. That goes there. This goes there. And as we mentioned, we need to specialize it. So to do that, I will cheat.
Is
And
it possible you can raise it up a little bit? I can see the words.
can everybody not see the word?
Yeah, I couldn't see like the bottom half of the word.
Okay, got it. All right, that's what I was fearful of. All right, so hold on.
Just curious, the font is this size just because you want to cover all the stuff in one slide, right?
Yeah, it's going to get ugly.
Got it.
All right, can you see it now?
Yes.
Yes.
Okay, excellent. So here is our skeletal muscle cell. And, whoops. This will be our motor end plate. All right. Now, as we know, the invaginations are one of the specializations of the motor end plate. The second specialization of our motor end plate, and I will draw them like this. I'll make it a little bigger. I'll just draw 3 of them here. Excellent. Although I need more space. Can you guys see the bottom of the cell now?
Not the bottom line, no.
Okay, but that's fine though. That we don't need to see the bottom line. I just need a little bit of space there. Excellent. And these are our, and I'm going to abbreviate this again for space. Chemically gated, so that's what CG stands for. Cation channel. Oops. All right, so that is those right there. Excellent. All right. So that is our skeletal muscle cell, its motor end plate, and its 2 specializations. Can we all see that?
Yes.
Okay, excellent. All right, perfect. So that's step 1. Step 2, we need our synaptic end bulb. I think we can make that work. And I need lots of space in between them. So I'm exaggerating the space in between them so that this would work. And then I need that up there.
Sorry, a quick question. The motor end plate, is it the 3 that you drew at the bottom?
No, the motor end plate is this entire region. So here, hold on. This entire region here is the motor end plate. And
Oh,
remember,
gotcha.
the motor end plate has the specializations to them. And then it also has 3 chemically gated cation channels. Now, obviously, the real one has far more than that. But again, we're just doing it for simplicity here. There. All right. Can you guys see the top of that line okay?
No.
You can't see the top line? All right, how about this? Can you see that?
Yes.
Yes.
Okay. All right, so this is the axon and the axon terminal. And then this here is my synaptic and bulb. All right, so we have that. Perfect. Excellent. We know our synaptic end bulb has some specializations to it as well. The first specialization, and I'll draw 5 of them here, are our vesicles. And these vesicles, again, contain acetylcholine. And again, I'm abbreviating these because it meets my need. So let's and perfect. Yay. Excellent. So we have our vesicles containing acetylcholine. We have mitochondria we know in there because this requires a lot of energy, but I'm not going to draw it because it isn't as important for this part of the process. But what we do need to draw is a couple of things. The first here, as we mentioned, are our, and again, to be consistent, oh, she'll just spell it out, voltage gated calcium channel. Which lets calcium into the cell. Because calcium makes cells do wonky things, right? Now, there's one more component we didn't talk about, but most cells have it. As we've mentioned, do cells necessarily want calcium inside of them? Do cells want to be doing wonky things all the time?
No.
No. So one of the things that most cells have is a calcium pump. Pump, remember, makes it an active transporter, which means it uses ATP to remove calcium from the cell. Okay? And again, most cells are going to have that the same way as we know most cells, and I'll go ahead and draw it down here for the fun of it. As we've also talked about, most cells have a sodium, potassium pump, which, as we know, moves 3 sodium out of the cell and 2 potassium into the cell. So these are things that all cells have, but it's the voltage gated channel that is important, is the specialization, but this pump's going to be important as well. All right. The last component, as we know, of course, is this space in between the two. Whoops, hold on, I don't want to do that. Is the space in between the two, and that space, as we mentioned, is the synaptic cleft. It's the space in between. And as we know here inside the space in between, let's draw it this way. I'll draw it as a little Pac-Man. And this Pac-Man is our acetylcholine esterase, right? Which again, as we said, once we do that, we can abbreviate it ACHE. which of course, as we mentioned, is the enzyme that is going to break down our acetylcholine in the synaptic cleft. All right? Questions on this? All we've done so far, there isn't really any new information on here. This is just the anatomy we have been talking about. So any questions on this? All right. If we are comfortable with the anatomy, then we are ready to start making this messy. However, as I said, if we are going to talk about how we initiate this process, how we terminate this process, and how we modify this process, there's two things we're going to need. First is we're going to need to be fresh-minded and fresh, so we need that. And second, I'm going to need caffeine. So let's go ahead, even though it's a teeny bit early because we got a late start, this is a good natural stopping point because, like I said, things are about to get ugly. So let's go ahead and take our first break. We'll take a 15-minute break, come back at 9:20, and at 9:20 we will restart from here. So restart at 9:20, and I will start the recording at that point. All right. So I will ask the question again before we get started. Any questions on any of this for the anatomy? Because that's basically what we have here. All we have here in black is the anatomy. So I want to make sure when we get a chance to go through all of this, it all makes sense because we're going to be adding layer upon layer upon layer upon layer to this. So I want to make sure that we are clear on this before we move forward. So last chance, any questions before we get started? All right, then let's roll. So our goal first, as I mentioned, and I'm going to be doing all of this in blue, is for initiation of communication at the neuromuscular junction. All right. Now, as I've been emphasizing, what, of course, is the first step in this process going to be? If I want to contract a muscle, what's the very first thing I have to do?
Communicating that neuro.
You're right. We want to communicate at the neuromuscular junction. We need to produce a neural action potential. But even before the neural action potential, if I want to move my finger, what's the very first thing I need to do?
Think about moving your finger?
Bingo. The decision. Absolutely. Because it is a conscious act, the very first step absolutely is the decision. You are correct. So the first step is definitely going to be the decision. We decide we want to contract the muscle. When we do that, though, as was mentioned, we are going to generate a neural action potential. Now, we have mentioned this, but I will emphasize it again. A neural action potential, an action potential is a big electrical signal. I think we've said that many times. However, what I'm not sure we've said, and I want to make sure we emphasize it, is a neural action potential is a big positive signal. It is a big positive signal. And it's like a wave. When we get to the nervous system, and this kind of is baby steps into the nervous system for him, we will see that these action potentials, electrical signals in general, move a lot like water. If I stand at the end of my pool, because after all, I'm a college professor, so, you know, I get that kind of scratch where I've got the massive Olympic-sized pool in my backyard. And because I have plenty of cars to spare, I stand there holding a massive car over my head, dump it into the pool, and I generate a massive wave. And when I generate that massive wave then spreads across the pool. And that's what we have here. We have a big positive signal, and that big positive signal is going to spread down the axon. And when it spreads down the axon, that, and now I can abbreviate it, neural action potential reaches the synaptic end bulb. We know, as we've talked about, that the resting, oh, hold on, let's see if I can sneak this. All right, I'm gonna have to cheat a little bit. I want to grab this and move it there. And then I think it's just easier to delete all these. Come on, and then just redraw them. Perfect. All right, so as we know, oh, let me move that too. Used to doing this on the board. I'm not used to doing this here on the computer like this, so it gets a little trickier. All right, so as we know, we have a resting membrane potential in the cell, and the resting membrane potential of the cell, what did we say it was again?
-70.
Excellent, -70 millivolts. Perfect. That is the resting membrane potential of the cell. All right. This big positive signal reaches our synaptic end bulb, and it makes the synaptic end bulb more positive. We have a fancy name for that. When we make a cell more positive, we say it depolarizes the cell. So our neural action potential reaches the synaptic end bulb, and it depolarizes the synaptic end bulb. And it doesn't just make it +1 or +2 or +3. This neural action potential is a massive positive signal. So it is going to depolarize the synaptic end bulb to threshold. And when our synaptic end bulb reaches threshold, what is the immediate effect of that going to be?
It's gonna open that door, right?
Exactly, perfect. When we reach threshold, the immediate effect of that is that our voltage gated, and I can abbreviate it now, voltage gated calcium channel is going to open. And what is the immediate effect of that? Once the door is open, what can pass through that door?
The calcium.
Yeah, calcium. And which way does calcium go? Into the cell or out of the cell?
Into the cell.
Perfect. So the immediate effect of the depolarization to threshold is our voltage-gated calcium channel opens, and that allows calcium to enter the cell. All right, so let's go ahead and draw that. Got an arrow. We'll make it blue, and that blue arrow entering into the cell, entering into the cell, and of course we got to say what's entering into the cell. So it is our calcium. Excellent. Now, remember, as we said, there is this pump, and this pump's job is to kick calcium out. It uses ATP. Hold on, I want that to be black. It uses ATP to kick calcium out of the cell. Kind of the same way that if you're in a little rowboat, one of the things that you often have in a rowboat when you go fishing is you have a little hand pump, so that if a little water gets into the boat, you can use that hand pump to pump the water out. However, if someone drills 17 holes into the bottom of your boat, is that little hand pump going to be able to get the water out faster than the water's coming in?
No.
No. And that's exactly what's happening here. While the pumps may be trying to kick some of the calcium up, there's a lot more calcium entering into the cell. And so what happens is we get an increase in calcium. Oops, and this I do want blue. So increase in the concentration of calcium inside the synaptic end bulb. All right. So even though there's that pump there, it can't keep up with all the calcium coming in these these voltage-gated calcium channels that have opened, and now there's more calcium inside of the synaptic end bulb. All right, you with me so far?
I am, yes.
Excellent. That's all. Excellent. And you will be the speaker for the class. Excellent. If it's making sense to you, then that'll work. All right. So as we talked about, calcium makes wonky things happen. In this case, what's going to happen is calcium is going to trigger the exocytosis of our acetylcholine. Notice my wording was very careful there. I said it triggered the exocytosis, right? Calcium doesn't grab the vesicle and bring the vesicle to the plasma membrane. In fact, there are 25 proteins involved in that process. There are 25 proteins that are involved in grabbing a vesicle, bringing the vesicle to the plasma membrane. When it brings the vesicle to the plasma membrane, it then fuses to the plasma membrane. And when it fuses to the plasma membrane, it opens up and it releases its acetylcholine into the synaptic cleft. So this is an incredibly elaborate process we could spend a whole week talking about, and we are not going to. But the key is that it triggers the exocytosis. It activates those 25 proteins, and those 25 proteins bring the vesicles to the plasma membrane. And the net effect of that is we release acetylcholine into the synaptic cleft. All right. Questions on that? Now remember, we have this acetylcholinesterase that can gobble some of this up, but we're releasing a lot more than these can gobble up. So we have a lot of acetylcholine in the synaptic cleft. Now, once the acetylcholine is in the synaptic cleft, is it just going to stay there hovering, shivering, scared up against the wall of the synaptic end bulb?
No.
No. What's going to happen is that our acetylcholine is going to diffuse across the synaptic cleft. And once it diffuses across the synaptic cleft, it is going to bind to the chemically gated cation channels. And of course, when it binds to the chemically gated cation channels, our chemically gated cation channels open. All right, so that there like that, that there like that, and let's draw some acetylcholine binding to our receptors. Now, as we mentioned, when the channel opens, what kind of channel was it again?
Chemically gated cation channel.
Exactly, cation channel, chemically gated. So we have the key, we've unlocked it, and it is a chemically gated cation channel, meaning cations are going to flow. And what were the two cations that we said we're going to move?
Potassium and sodium.
Excellent. And are potassium and sodium going to flow in the same directions? Which way is, if we open this channel, which way is potassium going to flow?
Outside.
Excellent. Remember, if we talked about its concentration gradient, we know there's a lot of it inside the cell, very little of it outside the cell. So potassium leaves the cell. And sodium, which way did we say it was going to go?
Inside.
Enters the cell. Excellent. So let's think about this. We have, I need some more arrows. On this side, we have sodium that is entering the cell. And on this side, we have potassium. And I'll cheat and then grab that and move that there. Leaving the cell. Now, this muscle cell is a cell. So of course, not surprisingly, it has a resting membrane potential. And what do you think the resting membrane potential of our muscle cell is? Not a trick question. Negative 70 millivolts. Excellent. Negative 70 millivolts. Now, if one sodium enters the cell and one potassium leaves the cell, I have one positive ion coming in, one positive ion leaving. Am I going to change the membrane potential of this cell? No, it's going to stay the same if one sodium came in and one potassium came out. However, with this cation channel, the movement of sodium and potassium, as it turns out, are not the same. Only a little potassium leaves the cell, but a lot, tons of potassium enter the cell. So while let's grab that, move that like that, and then emphasize this like that. So there is a ton, okay, that may be a little too big. So there we go. There is a ton of sodium entering into the cell and only the littlest bit of potassium that leaves. So notice there's an uneven movement of the cations. So what is the net effect?
It's gonna increase or
There you go, say it.
depolarize it.
Perfect. The net effect is it makes the inside of the muscle cell more positive, and you're absolutely correct. The fancy word we say for that is this is going to depolarize the motor end plate, right? And again, we'll put it in parentheses. It makes it more positive. All right? And it could just make it a little bit more positive, or it could make it a lot more positive. However, and this is where the key is, I want to make sure we fit this in here. So let's, I'll put it over here. And I'm going to increase the font just the slightest bit. If the depolarization is big enough, and how do you think we know if it's big enough? Might there be a magical point we might need to reach that would be critical?
The threshold.
Bingo. If it's big enough to reach threshold, if the depolarization at the motor end plate, let's actually add that in, at the motor end plate is big enough to reach threshold, then we generate a muscle action potential.
By the way, we can't see anything after. Okay.
Hold on. There, is that better?
We can only see like half of the second line. Right there.
Well, here, hold on. I'll sneak it in over here then, because I want to make sure we can see that. All right.
I'm cutting off some of the other text.
Well, hold on. So there. Can you see now?
Yep.
Okay,
Yes.
excellent. So here we generate, and I'll draw the drawing to emphasize this. We are going to generate our big muscle action potential. And remember, we talked about how this big positive signal is like water. So we generate our muscle action potential. And if we generate that big wave, is that big wave going to stay right there at the motor end plate? No, it is going to then spread down the sarcolemma. There we go. And then let's draw the arrow. There. Excellent. All right. So there you go. Just that simply, we have described the process of initiating communication at the neuromuscular junction. Questions on this process? All right. Well, I want to do this. I'm doing this for two reasons. One is obviously I want to make sure you understand this information. However, The other reason I think it's beneficial for us to take the time and go through this together is because in reading your essay questions, because I started your lecture exams, one of my concerns is, and one of the things that can lead to frustrations from a student standpoint, is when they feel they know and understand a concept, but they don't get a lot of points on that essay question. I know that can be incredibly frustrating, and it can be confusing because you feel you know it. However, one of the things that I really want to emphasize, and I think I tried to do that when we were going through this, but I want to do it again, is, as I've mentioned, the way you maximize your points is by giving me the specific details, being precise in your wording. And this is a perfect example. Notice if the first essay question on your lecture exam is to describe the process of initiating communication at the neuromuscular junction, you could very easily describe that as you make a decision to contract the muscle, your synaptic end bulb releases acetylcholine, sodium enters the muscle cell, and we generate an action potential. Everything I said there is 100% accurate and true. However, what you're lacking is the steps. What you're lacking is the specificity in the details. One of the things I tried to emphasize as we were going through this, as you're writing out these as essay questions, is what is the immediate effect of what you described, right? You make the decision to contract a muscle. The immediate effect of that is you generate a neural action potential, a big positive signal. That big positive signal spreads down the axon to the synaptic end bulb. The immediate effect of that is it depolarizes the synaptic end bulb. The immediate effect of that is the voltage-gated calcium channels open. The immediate effect of that is calcium enters the synaptic end bulb. The immediate effect of that is the calcium triggers the exocytosis of acetylcholine, where vesicles are brought to the plasma membrane and they release acetylcholine to the synaptic cleft. The immediate effect of that is acetylcholine diffuses across the synaptic cleft, binds to the chemically gated cation channel and opens them. A large amount of sodium enters into the cell and a small amount of potassium leaves the cell. The net effect of that is our muscle cell at the motor end plate is depolarized to threshold, and at threshold, our motor end plate generates a muscle action potential which spreads down the sarcolemma. Both those descriptions I gave were 100% accurate, and every statement in them was true. However, one of those had a lot more detail, a lot more specificity, and that is what is going to maximize your points. So I really want you emphasizing each step. Be specific, be detailed as you're going through these things. All right? Questions on that? All right. I've done it here. Let's look at the pretty picture from your textbook and go through it there as well. So again, here is our starting point. Whoops, wait, what is going on? All right, I'm not sure.
Will you save that picture in our lecture images too?
Yes, in fact, let's do that. I think I already saved the anatomy one. I thought I saved it already, but let me go back and I will save it again just to make sure. I know I saved the anatomy one.
That one was really helpful.
Yep. Well, trust me, it's going to get a lot messier. All right. All right. Need to go back to here. Okay, excellent. So let's talk about the process of initiation of the muscle action potential. Again, no new information. We're just going to go through it all again. Like I said, we're going to beat this dead horse. It starts with the decision. We decide we want to contract the muscle, and we produce a neural action potential in the brain. It is a big positive signal, and big positive signals make things more positive, and we have a big fancy word for that. It depolarizes or causes a depolarization. That depolarization spreads down the axon and reaches the axon terminal, right? Finally to the synaptic end bulb. The immediate effect of that depolarization of the synaptic end bulb is it reaches threshold, and that opens our voltage-gated calcium channels. Immediate effect of that is calcium enters the cell, and calcium triggers or causes the synaptic vesicles to bind, and again, you got all the pretty pictures here, to bind to the plasma membrane, releasing their acetylcholine into the synaptic cleft via exocytosis. That acetylcholine diffuses across the synaptic cleft and binds to the chemically gated, or remember, we can also use the term ligand gated, cation channel, that receptor. Here, if we look at it up close, that acetylcholine, remember, is the key that opens the door, but it's not what passes through the door. What passes through the door is sodium and potassium, but not equal amounts. Only a little bit of potassium leaves the cell, but a whole lot of sodium moves in. So we have a lot more positive things entering the cell than leaving the cell. The net effect is that it depolarizes the cell. And and there's the key word if, one of the big deals about these synapses is it allows decisions to be made, right? We'll use the example earlier of the light switch. When I flip the light switch, the light always turns on, right? There's no decision in that. The light doesn't decide, is today a good day for me to turn on? Am I tired in the morning? No, it's hardwired together. We don't want our nervous system to be hardwired together, right? Because again, you want to be able to make a decision. If I stare at you, holding my mouse in my hand, over Zoom and say, I'm going to kill you, and a bear with an axe walks into your room, looks you in the eye and says, I'm going to kill you, you are getting the exact same stimulus from both of us. But are you going to respond the same to both of us? No, of course not, right? Absolutely not. So we want to be able to make decisions. And so that's the key. If the signal is big enough, then we get a muscle action potential. So it's not hardwired together. We can actually modify the behavior. Yes.
So for the purposes of this process, are we just assuming that our signal will always be big enough, or will we see examples where it's not big enough and we cannot create a muscle action potential?
So in this case, if we are making the conscious decision to move a muscle, we are going to assume that it is successful. So in this process. Now, like I said, there is a third part where we will modify it, where we'll see that may fail. But if I ask you to initiate the process, then yes, the expectation is that you're going to be successful.
Okay. So
yep. Right. And again, I know we think of it in terms of conscious activity, but we've also talked about reflexes as well. Right? If you're sitting there at the doctor's office and hanging your legs off the side of the table, and the doctor hits you in the knee with a hammer, what happens to your leg?
It moves.
Yeah, your leg kicks out. Exactly, right? Everybody comfortable with that idea?
Yeah.
Excellent. What happens if the doctor then puts a sleeping baby right next to your foot and hits you in the knee with a hammer? Are you going to kick the baby?
Yeah.
Really? No. You can actually stop the reflex. Even though it's a reflex, you can actually stop that. Nobody's going to kick the baby, right? So absolutely not. So again, so we want to be able to modify behavior.
Got it.
All right, and that's what we can do here. All right, questions on that? Again, if this doesn't make sense, you need to let me know now, because it's about to get a whole lot worse. All right, then let us switch gears. Now that we have initiated the process, this again, notice our muscle hasn't contracted yet, but we are just focusing on step one. We're just focusing on communication at the neuromuscular junction. This, we know we've generated that muscle action potential. We know down the line that's going to cause our muscle to contract. Great, I'm contracting my muscle. But I also don't want this muscle to contract forever. Eventually, I want it to relax. So what we need to do now is talk about our second process. And that second process is, oops, hold on, I made it big again. This part can be big. Termination, right, of communication at the neuromuscular junction. So that is what we're going to talk about now, termination. And guess what the first step of termination is as well?
To think about
Last
it.
step of first step.
Yeah, once again, we're going to make the decision. We decide when we contract it. We decide when we are going to relax it. All right, so that part is the same. When we make the decision to relax the muscle, in this case, what happens is we stop generating neural action potentials. If we want the muscle to contract, we have to produce muscle. We have to produce muscle action potentials. We want to produce muscle action potentials. We have to produce neural action potentials. So once we decide to stop contracting the muscle, we stop generating neural action potentials. So notice now the effect of that is going to be there is now no new big positive signals spreading down the axon. Now, so there is no, whoops, no. Do that. No more neural action potential. No more big positive signal reaching the synaptic end bulb. Now that big positive signal caused our synaptic end bulb to depolarize, to get more positive. But if that wave goes away, is the cell going to stay positive forever? Well, remember, cells have a resting membrane potential where they like to be when they're at rest. If we stop messing with it by producing a neural action potential, guess where the cell's going to want to go back to?
Just the resting.
It's going to want to return to its resting membrane potential. So our synaptic end bulb is going to return to the resting membrane potential. And again, we have a fancy name for this. The fancy name for this is we say the cell repolarizes. So the cell repolarizes and goes back to rest. Rest. Wow, I didn't do that even close. Resting. There we go. Resting membrane potential. So the synaptic end bulb is going to return to the resting membrane potential. I think I can sneak that in there. That'll make sense. And the cell repolarizes. Notice when it repolarizes, it passes threshold on the way down. And guess what happens to our voltage-gated calcium channels when we go below threshold? Well, if going above threshold caused them to open, going below threshold caused them to close. Our voltage-gated calcium channel closes. Oops. So now there is no new calcium entering the cell. So big caps, no new calcium enters the cell. That solves half of our problem, but the other problem is there is still all this calcium inside of the cell. If only there was a way for us to get calcium out of the cell.
The calcium pump.
Ah, the calcium pump wasn't very effective before when those voltage-gated calcium channels were open, but now, with the voltage-gated calcium channels closed, oops, don't want it to be caps anymore. The pumps can remove the calcium from the synaptic end bulb. And when there is no calcium in the synaptic end bulb, there is going to be no new exocytosis of acetylcholine. So our exocytosis stops. And let's draw a little X's here just as a reminder of that. All right, So no new exocytosis, no new acetylcholine into the synaptic cleft. All right. So far, so good?
Mhm.
Excellent. All right. However, we still have kind of a similar issue to what we did before. And that problem is there is still lots of acetylcholine in the synaptic cleft. So we need a way to get rid of it. And in fact, there are 3 ways. The first way to remove is just simple diffusion. Our synaptic cleft is open. Yes, the cells are very close together. Yes, our motor end plate has those nooks and crannies, but there is nothing stopping an acetylcholine from just, all right, that's way too big again. There is nothing stopping that acetylcholine from just diffusing away. Right? If the acetylcholine, and I'll just go ahead and draw one of them, is over here next to the cell here, and there are no receptors there, can it have any effect on the motor end plate anymore if it's not in the motor end plate? No. So one of the ways we can get rid of it is it can diffuse away. The second way we can get rid of it is actually with endocytosis by the synaptic end bulb. This acetylcholine is a useful chemical signal that can be reused. So some of this acetylcholine is going to be bundled up, brought back into our synaptic end bulb, and can actually be put back into a vesicle to be used again. So there can be some endocytosis. That is going to occur. However, there is a third and bestest way of getting rid of it. And guess what the third way to get rid of acetylcholine is?
Is it to diffuse it through the cleft and bind it to the cation channels?
Well, but we don't, remember, we want the muscle to relax. We don't want it to bind anymore. We want to get rid of it.
Okay.
If only there was something like Pac-Man who could gobble up those little dots for us to help us to get rid of that acetylcholine.
There
Oh, wait.
is.
Don't we have something like that?
Mhm.
Yeah, that special enzyme. What did we call it again?
Acetylcholinesterase.
Perfect, absolutely. The third way that we can get rid of acetylcholine is to break it down with acetylcholine. And since I have it spelled out right next to it, I'm going to abbreviate it here so that it fits. The third way we can get rid of acetylcholine is to break it down with acetylcholinesterase. So using these 3 methods, diffusion, endocytosis back into the synaptic, that should be synaptic end bulb, not ESB. So synaptic end bulb, not ESB. There we go. Oops. Perfect. All right, and then little Pac-Man here is going to gobble up the dots. In this way, we're able to get rid of all of the acetylcholine that is in the synaptic cleft. And when we remove ACH from the synaptic cleft, and I'm just going to abbreviate that synaptic cleft, what happens to our chemically gated cation channels?
They close.
Okay. Are going to close. Excellent. And when they close, that means no new sodium into the cell. Now, again, it also means no new potassium out of the cell. But was it really potassium that was driving the process?
Not really.
No, it was sodium coming in. And thanks to the pumps, our muscle cell returns to its resting membrane potential. And what did we call that process again?
Repolarizer.
Excellent. Repolarizes, which again is just a fancy way of saying that it returns to its resting membrane potential. And when it returns to its resting membrane potential, there is no new muscle action potential. And we have stopped communication at the neuromuscular junction. And just that simply, we have terminated the process. Questions on that? All right, let's look at it again from your textbook. So again, remember, the key to stopping the muscle action potential is we have to get rid of the acetylcholine. Get the acetylcholine out of the synaptic cleft. Well, to do that, the first thing we have to do is stop releasing acetylcholine from the synaptic end bulb, right? So our synaptic end bulb has to repolarize, right? We stop producing neural action potentials. When we stop producing neural action potentials, we stop depolarizing the synaptic end bulb. Our voltage-gated calcium channels close. No new calcium into the synaptic end bulb. We pump the calcium out, and once the calcium leaves the synaptic end bulb, no new acetylcholine is released from the synaptic end bulb. However, as we also talked about, whoops, oh, wrong direction. We have to get rid of the acetylcholine that's there. Some of it's going to diffuse out. Some of it's going to be broken down by acetylcholinesterase, and some of it is going to be removed by endocytosis. Once we get rid of the acetylcholine, our chemically gated cation channel closes. No new sodium can enter the cell. The muscle cell repolarizes, and our muscle action potential stops. All right. Questions on that? All righty. So we've initiated communication at the neuromuscular junction. We have terminated communication at the neuromuscular junction. To truly show that we understand this process, one of the things we want to do is be able to modify, mess it up, all right? If you know how an engine works, then one of the things you can do is know that if you remove, right, if you, 2 or 3 of the spark plugs, right, that's going to cause problems, right? And the engine's not going to work properly anymore. So understanding how it works helps us to understand how we can manipulate this. So let's do that. The third thing we need to understand is how to modify, or more importantly, how modifications can affect this process, and ultimately, what effect it has on the muscle cell. I'm going to give you 3 examples. The first example we will use will be the example of Botox, botulinum toxin. Where do you, whoops, that's way off. Where do you typically find botulinum toxin?
In expired cans.
Yeah, right, spoiled canned goods.
Also in Botox.
Yeah, we'll get to that one in a second, but yes, it started in spoiled canned goods, absolutely. We started with that botulinum toxin that is found in spoiled canned goods. It turns out what botulinum toxin actually does is it is a toxin that is produced by a bacteria, and basically what that bacteria does is it breaks one of the synaptic end bulb proteins. Oops, proteins. It turns out that protein happens to be called SNAP. You don't need to know that name. Remember how I mentioned the process of exocytosis requires 25 proteins to be involved in it? Well, all botulinum toxin does is break one of those proteins. But it turns out it's a pretty important protein. That protein, when broken, stops the vesicles from being brought to the plasma membrane. So let's think about how this would affect the cell. Once again, we are going to make the decision, right? We are going to generate a neural action potential. We are going to depolarize our synaptic end bulb. Oops, depolar synaptic end bulb. Our voltage-gated calcium channels open. Calcium enters the synaptic end bulb. And then what happens?
We try to use the pump to remove some of the calcium.
Well, but again, remember, what botulinum does is breaks that protein. So now what happens is, even though, oops, wrong button. Even though calcium is coming into the cell, calcium can't get the vesicles to bind to the plasma membrane. So what happens is, with that broken protein, stops the exocytosis of acetylcholine.
Okay.
So that means there's no acetylcholine in the synaptic cleft. And if there's no acetylcholine in the synaptic cleft, can we open our cation channels?
Oh.
Can we allow sodium into the muscle cell?
Because our
Can-
cation channels won't open.
Right, cation channels would open, so sodium can't come in. Can we depolarize the muscle cell to threshold? Can we get a muscle action potential?
No.
No. So notice the effect of this is there's going to be no muscle action potential. So even though I'm telling my muscle to contract, can the muscle contract?
No.
No. What this is going to do is produce what we call a flaccid paralysis. So we tell the muscle to contract and it can't contract. Now, as someone mentioned very cleverly, we find botulinum toxin in spoiled canned goods. It is a toxin produced by a bacteria. But botulinum toxin sounds so negative. If we give it a cutesy name like Botox, then people have no problem injecting it into their foreheads. Because after all, as we'll learn, we have a nice big muscle here on the front of our forehead. And muscle tone in that can, as our skin becomes less elastic as we age, can cause wrinkles to occur. However, if I inject that toxin into these muscles, it breaks that protein, that muscle becomes completely flaccid, and yes, I can no longer show facial expressions, but my forehead is nice and smooth. And as we know, the most important thing in the world is to look beautiful. So who cares if you can't express your emotions anymore? As long as you look beautiful, that is all that matters. And so they'll have big parties where they get all their friends together, right? Back in ancient times when we could have big parties, you get a big party, get a doctor there, a bunch of bottles of wine, right? And everybody be injecting everybody into their forehead, having a good old time so they can look young again. But here's the problem. What happens if you inject that botulinum toxin a little too deep and it gets into the blood? And once it gets into the blood, it can go everywhere. Like, I don't know, maybe a muscle like your diaphragm. What do you use your diaphragm for again?
Yeah, that's your lungs, right?
Yeah, for breathing, absolutely. So if you break the SNAP protein, break that protein in the neuron that's communicating to your diaphragm, and you can't contract your diaphragm anymore, right?
Can't
How's
breathe.
breathing going to be for you? Is that going to be a fun experience?
No.
No. Now, again, the neuron is a cell. So cells have the ability to produce new proteins. Of course, what happens is first the signal has to get back to the neuron to tell the neuron that the protein has been broken. Then the cell has to make the protein. Then the protein has to ship that protein back to the synaptic end bulb. And depending on the cell, the synaptic end bulb, the cell going from your spinal cord down to your diaphragm, the axon, typical transport is about 1 inch a day is how long it takes a protein to be transported down your axon. How long can you hold your breath for? Long enough to get that signal to there and then send the protein and fix the protein and do all of that? No, of course not, right? So you're basically being artificially ventilated until you hopefully recover from them. But like I said, because we understand it, once we understand the mechanism of how this works, we can use it to their advantage, right? Whether it's cosmetically to make those wrinkles go away. If you have excessive sweating, again, notice it blocks exocytosis. So one of the things that can happen is if you have excessive sweating, they will inject botulinum toxin into the sweat glands, into those eccrine sweat glands, blocking the release of the sweat, decreasing the amount of sweat that you produce. Right? One of the things that we learn, one of the reasons we learn about the body is the more we learn about the body, more we want to be able to manipulate it. And this is a great example of that. All right. Questions on botulinum toxin? All right, let's talk about a second example. We'll stick with green, but I'll use a different color green. Let's use this one. Actually, let's use that one. The next toxin I want to talk about is curare. Anybody know where you get curare from? It's a toxin that can be found in some poisonous frogs. Oops, I spell frogs right. Some poisonous mushrooms. You may not have heard it by name, but anytime you've seen any good adventure movie worth its salt, at one point or another, the protagonist and all of its assistants are running through a jungle, being chased by natives, and all the natives are using all these blow darts to throw these darts at the people. And of course, all the assistants of our protagonist are getting hit and stumbling to the ground while he is able to avoid all of it and get away. Curare is another toxin that causes a flaccid paralysis, causes the muscle to not contract, but it does it in a very different way, right? Remember, we talk about these the chemically gated cation channel. We keep using the example of acetylcholine being the key that unlocks the door, right? And again, normally we're doing this in the classroom. So I mentioned how when we get there first thing in the morning, I use my key to unlock the door and we come inside for a lecture. But what if during the night, someone had put gum inside of the lock? If someone shoves some gum inside of that lock, am I going to be able to get my key in there and open the door?
No.
No, and that's what curare does. Curare binds to and blocks the chemically gated cation channel. So what happens is, and I'll use it as a nice green block here, it binds, oh no, not dark green, I want the light green. It's going to bind to and block our chemically gated cation channel. Gums it up. So now, again, we walk through the process. We make the decision. We produce the action potential. It spreads down to the synaptic end bulb. It depolarizes the synaptic end bulb, opens the calcium channels. Calcium rushes in. Calcium triggers the exocytosis of acetylcholine, and we get all this acetylcholine in the synaptic cleft, but with that gum in the lock, our acetylcholine cannot open the door, open the receptor, can't open the channel. And if it can't open the channel, sodium can't come in. Sodium can't come in. We can't generate a muscle action potential. And again, we get a flaccid paralysis. There we go. You can dump a whole bucket of acetylcholine into that synaptic cleft, and is it going to make a difference?
Nope.
No, because if you gum up those receptors, then they can't function. It can't bind to them. So again, the muscle, we tell the muscle to contract, but it can't contract. However, again, remember, as I mentioned, once we understand these things, we can use them to our advantage. Notice the botulinum breaks a protein. It damages it. So when you put that into your forehead, it causes that muscle to become flaccid and it's nice and smooth. However, as we said, is your forehead going to be smooth forever?
No.
No, eventually that neuron repairs itself and the muscle tone comes back. And so every 3 months or something like that, you have to have your Botox re-injected when you're doing that. Notice this one just gums up the hole. It doesn't damage anything. So once again, by appreciating this, this causes the muscle to be flaccid, but it doesn't actually hurt the neuron and it doesn't hurt the muscle cell. One of the things that now all you hear about in the news is the coronavirus, but back in ancient times when the coronavirus wasn't around, there was a lot of tremendous interesting research going on, and a lot of that was going on with transplant surgeries. People have transplanted faces, people are transplanting hands, all of these types of incredibly, really delicate surgeries. And probably one of those face transplants, they're probably one of the greatest examples. You are taking this incredibly delicate skin and connecting it to these massive numbers of blood vessels and nerves and muscles. And the problem is, as we've talked about, muscles give us tone, right? When I'm not moving my face doesn't just slouch down off the surface of my skull because I have muscle tone. And the reason I have muscle tone is your muscles are making tiny little contractions that are helping to maintain the tone of your muscles. It's why your shoulder doesn't fall out of socket. It's why your face doesn't droop off your face and all those things. And it's not an issue for normal everyday life. But if you're in there delicately trying to connect a blood vessel or a nerve or the skin, even a small contraction can damage the skin, can damage what you're transplanting. So what they'll do is they will bathe the area that they're working on with curare. That completely paralyzes the muscles in those areas. They can make all the tiny little connections that they want, and then they just wash it out with saline. And once they wash the curare out, everything goes back to its normal function again. So it's a really important paralytic that is often used in surgeries for very, very delicate surgeries. All right. Questions on that? All right, I got one more for you. Let's change the color one more time. The last one, and this was one that was actually mentioned on a previous class, tetanus. Where do you get tetanus from? Rusty nails, excellent. Rusty metal. Tetanus, again, is a toxin produced by bacteria associated with rusty metal and things along those lines. What's interesting about tetanus is similar to curare in that it binds to the cation channels. However, in this case, unlike curare that binds and blocks it, tetanus binds and activates the channel. So what happens with curare is curare will come in, come on. Curare will come in, oh, that's supposed to be light green. There we go, lime green there. It will come in and it binds with the channel. And when it binds to the channel, it opens the channel. It activates the channel, meaning that the channel won't close. So what happens is you have sodium constantly entering the cell. And if you have sodium constantly entering the cell, you're constantly producing a muscle action potential. Muscle action potentials. And if you're constantly producing muscle action potentials, guess what's happening to your muscle?
Always moving, contracting.
Yeah, it's in a constant state of contraction. Right? It's what they call a rigid paralysis. right? My muscle's in this constant state of contraction where I can't get the muscle to relax. I can't move it. I'm locked in this position. In fact, that is what tetanus is commonly referred to as. What do they commonly refer to it as? Lockjaw, right? You've ever heard of that? Lockjaw? Because the muscles in your jaw contract constantly and you can't open your mouth. You think my lectures are bad now. Just imagine what it would be like if I had lockjaw and you had to listen to me talk like this all day long, right? You look in the old textbooks and you see some of the pictures of these individuals dealing with tetanus, and their bodies are just contorted into these excruciating looking conditions and positions from these muscles that are constantly contracting. Right? They're locked up in this position and it's painful and it's damaging, right? Remember, as we talked about, if you have that flaccid paralysis to your diaphragm, I can ventilate you and allow you to breathe pretty easily. However, if all the muscles of your thoracic cavity are all contracting at the same time, is it going to be easy for me to ventilate you when all those muscles are contracting? No, it's a very damaging, very painful, very dangerous condition. Which is one of the reasons why we get vaccinated against it, because it can be so devastating and dangerous. So there you go. Notice we have now talked about and identified 3 ways that we can modify communication at our neuromuscular junction. Right? We've initiated it, we've terminated it, and we've identified 3 different ways that we could modify it. Questions on that? All right, excellent. Well, just that simply, that is everything that I wanted to do from a lecture standpoint for today. All right, so what we get to do now is we get to switch gears. We will switch gears and move into our lab portion. Hopefully you have your red and blue pens. Hopefully you have all of your printouts and handouts ready. Although there is a little bit more anatomy I want to do before we get to that, but I think let's give some time to let this sink in. So let's go ahead and take our second break here. What time is it? It is 10:21, so let's come back at 10:36, and at 10:36 we will pick up from here. So we will restart.
I wanted to ask a question that was off topic.
Yeah.
We were talking about troponin, and troponin is basically the way to keep the cell resting in the muscles.
Yep.
So how would that relate to a heart attack? Why is there increased troponin after a heart attack?
I have no idea. No, I'm just kidding. The reason is it is a different type of troponin. The type of troponin that is found in skeletal muscle is different from the troponin that is found in cardiac muscle. So when cardiac muscle, really when all muscle is damaged, what happens is proteins are released from it, because remember, they're chock filled with proteins, and proteins are released into the interstitial fluid and get into the blood. So in the case of when cardiac muscle is damaged, troponin happens to be, the troponin in cardiac muscle is slightly different from the troponin in skeletal muscle. And so when that is released, it's something that can be looked for in the blood that is specific and unique to cardiac muscle that tells us that there's been damage to cardiac muscle.
Okay. And we'll go over cardiac muscle at a different date.
Yeah, at the end, we will. So what I will say is we will briefly go over it in this class, but remember when we get to the cardiovascular system in 431, we'll go into it in much more depth. In fact, if you take me for 431, we actually start 431 with the cardiovascular system. So the very first thing we'll talk about is the heart. So we will briefly touch on it, but we won't get into it as much in this class. We're going to focus primarily on skeletal muscle.
All right. Well, thank you.
All right. Excellent. That was a great question. Yes, I appreciate the confusion of that. But that's the short answer as to why that is. All right. All right. Again, let's take a full 15-minute break. So come back at 10:38, and at 10:38 we will pick up from there. Any other questions? All right. I'll see you back here in 15 minutes. Before we dive straight into our lab stuff, there's a couple more things I wanted to cover. Hopefully you guys saw that yesterday after class, I actually added a couple more images to that handout of the models from class. So I wanted to start by going over that a little bit first. So the first thing that I wanted to show you, some of the models from the classroom involve, so I need that, just the model tissue, muscle tissue models. Which type of muscle tissue do you think this one here is? You guys can hear me, right? Yep, excellent. Skeletal muscle, excellent. We have multiple nuclei, we have striations, they're long and cylinder shape. On the exam, I could show you histology picture of this, things along those lines, but I could also show a model like this as well. And remember also, one of the questions that I could ask is not just to identify this tissue type, but to give me some of the characteristics. So why don't you guys give me some of the characteristics you know about skeletal muscle tissue? Can be obvious stuff that you see when you look at it, or it could be excellent. So one of the things is it's multinucleated. Perfect. What else? Voluntarily controlled. Excellent. Give me another one. Long cylinder shapes. Excellent. Right. Cylinder shape. Excellent. Medium amount of regeneration. Things along those lines. Excellent. Perfect. What about this one? What tissue type would this be?
Smooth.
Smooth muscle. Excellent. Right. What are some of the characteristics we know about this one? What do we know about smooth muscle?
Spindle shaped?
Spindle shaped, excellent. Involuntarily controlled, unstriated, highest
One-
rate of regeneration. Yeah.
One nuclei.
Single nuclei, right? You know, one of the pieces of information you guys have missed from both of these is where they're located, right? Skeletal muscle is attached to the skeleton mostly. Where do we find this one?
Smooth muscle in the veins, arteries, and digestive tract.
Yeah, walls of the hollow organs, right? Absolutely. Those are good examples, but if we wanted to also give a general description, we could say it's in the walls of the hollow organs. Excellent. And so then, not surprisingly, what might this one be?
Cardiac muscle?
Cardiac muscle. And what are some of the characteristics we know about it?
Can have
Striated,
multiple, but
uninucleated, autorhythmic, involuntary, right? Absolutely.
Found only in the heart?
Only in the heart. And just out of curiosity, what is this black line, do you think? What do you think these black lines indicate?
Do we say the intercalated discs?
Intercalated discs. Excellent. And what type of cell junctions did we say we would find a lot of in these intercalated discs? Desmosomes and gap junctions. Perfect. Excellent. Desmosomes help to hold the cells together, and the gap junctions, remember, allow them to rapidly communicate so they all work together as one. All right, so again, for these tissues, I could show you models, I could show you histology slides, ask you to identify them, ask you to identify characteristics like this. What is this? What is this? What are these? Things along those lines. But also you need to know the characteristics of these tissues type as well. That is definitely questions that I could ask on the lab exam. All right. This was the model we looked at first, right here. So again, this is from a front view, and then we looked at it from the top. The nice thing about this one is this one does a really good job of showing the different proteins within our myofibrils, all the myofilaments. Remember, we saw it cut at different regions of the organelle. And then what I found while I was doing that is also notice we see this nice view from the top. And I think from the top, we can see these areas a little bit better. So what region of the organelle did we say this one here is? Well, what proteins do you see on this cross-section of the myofibril? All right, this is just myosin. So if we have a cross-section through the myofibril and we just see myosin, what region of the organelle is this? H zone. Perfect. If we just see actin, like here or here or here or here, what region of the organelle would this be where we just see actin?
Z disc.
No, not the Z disc, it would be the I band. You're right, in the center of it would be the Z disc, but if we're just seeing actin, then that would be the I band. When we see actin and myosin, that would be the A band. And this one here, where we see the myosin all interlaced with myomesin, this would be the, what region of the organelle? M line, excellent. Notice that's exactly how I'm going to ask the question on the exam. I could have this exact picture with those exact stars in these locations saying what region of the organelle is this, or what proteins these are, so make sure you can identify those. Oh, and then notice now, excellent, because we had a chance to talk about it today, we also have that picture we were looking at before of the neuromuscular junction. Notice now with this illustration, we can actually see the parts. Here in yellow would be the synaptic end bulb, or orange, pardon. Orange would be the synaptic end bulb, and the yellow greenish dots, what would those represent? Vesicles. And what would these vesicles contain? Acetylcholine. Excellent. Right? Notice here in white, we see the massive invaginations of the sarcolemma. What specific region of the muscle cell would this then represent? Motor Endplate. And notice the pink thing in between, even though they got it kind of bulging out, really represents a space. And what would that space be?
Synaptic cleft.
The synaptic cleft. Excellent. And as we know, the big specialization is there is we have that acetylcholinesterase enzyme floating around inside there. Notice we can see the triad really nicely with this one, with the transverse tubule, the terminal cisternae, the sarcoplasmic reticulum. And notice from pink to pink, we can see one whole sarcomere for this view as well. So this gives a lot of good detail on that. There is a second skeletal muscle cell model in the class. This is it here. Notice again, we can see the plasma membrane. One of the things this one does really nice is it does a really nice job of showing the transverse tubules. Notice if we look at it from the top, we can actually see they've opened it up to show you that it's a fluid-filled tube that comes through it. However, they don't have as much detail to the myofibril. Notice they do a nice job of showing us the A band and the I band, and then the A band and the I band. And they kind of infer to the Z disc in the center. And I don't know if this is supposed to represent the M line or the H band, but it isn't quite as nice. But notice they've done a really good job with the transverse tubule and the sarcoplasmic reticulum and the terminal cisternae and the triads. And notice also they have a really nice neuromuscular junction. And here they've done a better job of making the synaptic cleft actually look like a space. So that's really nice as well. So I have these for you here to help you to be able to identify these things. Lastly, as I mentioned in class, we also have this great sarcomere model in the class. Here we see, well, actually, let's start with this picture here. Here we see the model in its entirety. And notice one of the cool things around it is had this little piece of plastic that wraps around it. If we zoom in on that plastic, we can actually see what it represents. Here in green is representing the sarcoplasmic reticulum that wraps around the myofibril. Here, the large part would be the terminal cisternae. Yellow would be the transverse tubule. And so notice the transverse tubule and the two terminal cisternae would represent that triad. So that's kind of what they're trying to represent there, is how the sarcoplasmic reticulum and the transverse tubules wrap around the myofibril. But if we remove that, and let's actually start with this picture first, we can see a sarcomere. Sarcomere goes Z disc to Z disc. Attached to the Z disc, we have our actin filaments. Here in the center, in blue, are our myosin filaments, the thick filaments, and then this represents the myomesin. If we look up close, notice that on our dimer, that is the actin, we have that thin yellow line representing the tropomyosin, and then the green handle, the troponin, that is going to move it out of the way. And here you get a closer view of the myosin heads represented here in blue. Then I'm going to grab on that actin and pull. So again, this here would be the H zone. This would be the M line. This would be the A band. This would be the I band. This would be the Z disc. So we could talk about the regions on this as well. And here it's pulled a little further out. So again, you can see those myosin heads a little bit more clearly. So these are some really nice models that are in the classroom. So I've made these available so that you can study them, look at them, and these are definitely things that could be on the exam. The other thing I've put in here, because I think it's a really nice model, and remember something we're going to talk about a lot, but not today, is, oh, and there it's pulled out more so we can see that. We're going to talk about the bones, and here is the scapula with the muscles. There are 4 muscles on the scapula that we are going to be responsible for, and so I want this model on here. I put it on here so that it would be, again, it's another important model in the classroom, so I just put it together so you don't have a whole bunch of different files to look at. But we'll save that and talk about that when we get to the arm. I guess that we'll start the arm on Friday. All right, so great resources that are there. So I've made that available. I changed the handout that was there so it wasn't just the one model, but all the stuff that are there. And then, like I said, hopefully what you have for today is your origin insertion handout and a red and blue pen. And then, of course, you should also have, well, let's change the view of this. Your study guide for this as well. All right, so any questions before we get started?
Quick question.
Yes.
So you know how we were all separated into groups and we had to come up with the activities to help us remember?
Yep.
Just to clarify, we only had to come up with one of each of those, right?
Correct. And
Okay.
technically, I'm not going to call on each group for every muscle. So actually,
that's what I was trying to figure out. Yeah, okay.
So group 1, actually, so thank you for reminding me of that. Group 2, got to make my list.
Yeah, because I didn't know if I should get a couple different activities ready in case every group was getting called, but you said no already.
So yeah, what I will do is I will randomly pick a group, and then we'll just go through. And so I'll say, all right, so for the masseter, it's going to be group 4. And then, you know, and so each time, so at the end, we'll do that. And so, so if yours is the same or yours is different, that's great. If you have one you really, really like and I pick a different group, then I'm happy to let anybody else who wants to share. But because again, like the goal of this is to help students to understand this material. And if you've got a really good mnemonic to help you that, and actually I have some favorites that I've come across in the many, many years. In fact, one of the reasons I do this is in one of the first years I did this and I had a student come up that I was just gobsmacked by the example, and it was so perfect and was so hilarious that I use it every single semester. So I will share it with you when the time comes, but still 12 years later, I'm still giving her credit for it just because normally, you know, once after a couple years, I steal something as my own, but she still gets credit for this because it's just that hysterical. So we'll get to that when we get to that. But yes, thank you. I got my list. So yes, we will do that. But yeah, it would take too long if every single group demonstrates it. So we'll just do one demonstration for each. And again, remember, you have to present it. And again, by just stating it and explaining it to us. But if you come on camera and demonstrate it for us, then you'll get a extra point, a bonus point of extra credit for doing that. And remember, only one person in your group has to be bold and brave enough to do that. But I'd like us to mix it up. All right. Any other questions? All right, so let's talk about the muscles of the body. As I mentioned, one of the things we're going to be responsible for are the origins and the insertions, but I want a better clarification of what exactly that is. So let's start first by drawing a couple bones. And we'll use yellow for the bones. So there's bone 1, and here is bone 2. Typically, when a muscle connects, and we'll go ahead and use brown for the muscle. Make this big. A muscle typically is going to connect across one or more. So a muscle attaches one or more joints, right? But you think about it, makes sense. If you had a muscle that just connected from one part of the bone to another part of the bone, when you contracted that muscle, would the body move in space at all? No, it wouldn't have any effect. So typically we need to have a muscle move across one or more joints. Now, in this case, when the muscle contracts, muscles get shorter and the two bones move towards each other. But when you contract that muscle, typically what happens is both bones don't move and meet in the center. Typically what ends up happening is that one end stays stable, right, unmoving, and the other end is pulled towards the first. So, towards the first. So what happens is when this particular muscle contracts, what would happen is this end would be pulled towards that end. And what would happen is that one bone would move and the other bone wouldn't. When we do this, typically the stable unmoving end is what we refer to as the origin. And the movable end, the end that moves, we refer to, be consistent, as the insertion. All right. So that is a pretty simple, pretty basic, pretty straightforward definition, which of course, because this is A and P, you should know right off the bat, isn't always going to be the case. And that's one of the frustrating things about this. But for the most part, this is how we define an origin and insertion. When we define an origin and an insertion, origins are going to be the fixed end. Insertions are going to be the movable end. Oh, and of course, hold on, let's go back. Undo that. Remember, as we have also talked about, typically the attachment point that is stable, the one that is the origin, we always use red to refer to, and the one that is the insertion and moves, we use blue to refer to. So origins are red, insertions are blue. So as you look at some of the models, some of the materials that are online, you'll see bones with these red and blue indications on them, and those are the origins and the insertions. All right. So again, by convention, this is colored red. And by convention, and remember, convention just means it's a rule we've made up, colored blue. Excellent. So that's how we primarily define our origins and insertions. However, there are some rules. Typically, and again, typically means mostly, but not always, origins are either closer to the midline of the body or more proximal on the appendage. So that is typically the case. Typically, origins are larger attachment points and insertions are smaller attachment points. And there's one other big issue. Bicep brachia is a muscle located in the brachial region of the body. But why is it called the bicep?
Because there's two origins?
Yeah, well, the reason it's called the bicep is bi meaning two, sep referring to like cephalon, head. It has two heads or two bellies to it. But you are correct. If you have a muscle like the bicep brachia that has two, the tricep brachia that has three, if you have muscles that have multiple bellies, multiple heads to them, each head has its own specific origin. So the key to this is with multiple heads, each head has its own unique origin. This is going to be very important on the exam, because on the exam, when I point to the bicep brachia, I'm going to point at one of the two bellies. Anyone know what the two bellies of the bicep brachia are called? They have very technical names.
Bicep belly 1 and bicep belly 2?
Not quite that basic, but pretty darn close. Long and short. We have a long head and we have a short head. So on the exam, if I point here and I point here and I ask you to identify that muscle, you can't just say bicep brachia. You also can't say the long head and the short head of the bicep brachia. You need to be specific. In fact, that is going to be one of your biggest key words on this exam. When you see on the exam the term be specific in your answer, that should cause warning bells to go off. because basically I'm asking for one of two things. I am likely asking for a specific belly or head of a muscle, or the specific origin of that specific head. On the exam, and I know we're getting a little ahead of ourselves here, but this is a good point to bring this up. As I mentioned, I have two heads to the bicep brachia. The long head, which happens to be lateral, that's how I remember it, long is lateral, short head, which is medial. On the exam, if I point here and ask you to identify this muscle, be specific, and you see bicep brachia, you will get it wrong. If you say long head and short head of the bicep brachia, you will get it wrong. If I say be specific, then guess what you have to do? You have to be specific. So you would say that is the short head of the bicep brachia. Or I could ask you identify the origin of the muscle, be specific. The origin of the long head is the supraglenoid tubercle of the scapula. The origin of the short head is the coracoid process of the scapula. So if I point here and ask you to identify the origin of this muscle, be specific, you need to say the coracoid process of the scapula. If you wrote in the space there, the origin of the long head is from the supraglenoid tubercle of the scapula, and the origin of the short head is from the coracoid process of the scapula, all of that is 100% correct. All of that shows tremendous knowledge. However, were you specific? No. And if you're not specific, you're not going to get credit for it. To get full credit, if you see the magic phrase, and again, my goal is not to be tricky. This is the specific phrase I will say. Be specific. If you see be specific, you know you're looking at something that has multiple heads, and you need to be very precise in your answer, specific to the one that I am pointing at. Okay? The good news is when you have multiple headed muscles, while they have different origins, they all have the same insertion and the same actions. So what that means is that I won't have to, it doesn't matter if I point here or here, insertion is the same. If I point here or here, actions are the same. What's different is name, what's different is origin. All right? Can't make the warning any clearer than that. I will give you that dead giveaway term, be specific. And because you're sophisticated students, you'll see that be specific and you'll know exactly what's going on. We're talking about something that has multiple heads, and you have to be precise with which one we are talking about. All right? Questions on that? All right, we have two more issues that we need to cover. As again, the key word on this was typical, because that's usually the case. But one of the troubling issues that we have is that muscles, the body, is more complicated than this. For instance, one of the things we're going to be talking about today is a muscle called the sternocleidomastoid. It's one of your neck muscles. One of the actions of this neck muscle is to flex the vertebral column, bringing the head down. It's able to do that because the origin is here on the clavicle, and it inserts into the mastoid process. So when it contracts, we bring the insertion towards the origin, and it brings it down. However, this isn't the only muscle that attaches to the head. So what can happen is I can use other muscles to stabilize my head in place, so that when I contract this muscle, instead of my head going down, what happens is my chest will come up. So what can actually happen is the origins insertions can actually reverse. Some of that happens in the neck. Where it's really common is in the leg. In the leg, whether your leg is extended or whether it's flexed, abducted or adducted, the origins insertions can actually switch. So like most things in this class, everything is more complicated than we're going to talk about it. But what we say are the origins, we're going to call the origins no matter what. What we say are the insertions, we're going to call the insertions no matter what, even if they sometimes reverse depending on the actions that they cause. All right? Questions on that? All right, there's one more piece of business we need to take care of from a vocabulary standpoint. And from a vocabulary standpoint, there we go, we need to talk about muscle groups. First thing we need to do is define what a muscle group is. The most obvious definition of a muscle group is it is comprised of 2 or more muscles, right? That makes sense. You can't have a muscle group that just has one muscle in it. But there are 2 other keys to the definition of the muscle group. They must be similar in their actions. Notice I didn't say identical. They have to be similar. Oops, what's going on? Similar, oh, there we go. In their actions. Not identical, but they have to be similar in their actions. And the other rule is they must share at least one attachment point. It can be either the origin or the insertion, but they must share at least one. There, can you guys see all of that? Yes, no, maybe. You guys fall asleep already? Can you guys see all of that? Yes or no?
Yes.
Excellent. All right. Excellent. So must be similar in their actions, not identical, but similar in their actions, and they must share at least one attachment point. It can be either the origin or the insertion, and in some cases, it will actually be both. Okay? Now, if you look at your study guide, there are some muscle groups that you only need to know the group.
We can't see that last.
Right, Ellen, I know, I gotta pull it out. There you go. Can you see it now? Can you see it now?
No.
No, okay, then I will bring it up here. How about there? Some muscle groups, you only need to know the group. For instance, as you look at your handout that should be in front of you, your study guide, you should notice about, oh, I don't know, I think it's 3 or 4 muscles down, the zygomaticus. The zygomaticus is a muscle group made up of 2 muscles, the zygomaticus major and the zygomaticus minor. Do you need to be able to distinguish the zygomaticus major and the zygomaticus minor on the exam? Do you see those names on your study guide anywhere? No.
No.
So you do not need to know them. You just need to know them collectively as a muscle group. The scalenes is made up of 3 muscles. Do you need to know what those 3 muscles are?
No.
No, you just need to know them as a muscle group. All right, so with those muscle groups, we'll just consider a muscle, and we won't worry about the different muscles that actually make it up. However, as you scroll through your study guide, maybe to the leg, you see that there are 3 muscles that collectively form the hamstring muscles. Do we need to know the 3 individual muscles that make up the hamstring?
Yes.
Yeah, anyone know what they are?
No.
Well, you've got your list in front of you. You should be able to read it off.
I'll read it off, yeah.
Quadriceps.
Quadricep is another muscle group, and for that one, we'll need to know all 4 of those muscles. But what are the 3 muscles that make up the hamstring?
Biceps femoris.
Biceps femoris,
And then I can't say the last one.
semimembranosus, and semitendinosus. So for those, you need to know them collectively as a muscle group, but you also need to know the individual muscles. And again, I've tried to make that as straightforward as possible. The ones that are just muscle groups that you need to know as a muscle groups, it says muscle group under them. And I didn't even give you the names of the individual muscles. The ones that you need to know individual muscles as well as the groups, I've provided you with both of those pieces of information. So again, as we go through this, I'll make a point of emphasizing that, but I want to make sure we understand what a muscle group is going into it, because we're going to run into a couple of them today. All right? Questions on any of that? All right. One last piece of business we need to start on before we get into our groups today, and that is naming the muscles. Part of the challenge in this class is determining the names of the muscles. And in general, there are 3, maybe 4 rules. 3 general rules that are used to name muscles.
Yeah, it is what it is. It's Greek would probably be.
And I think this is too large, so let me lower that a little bit. Excellent.
You know what I mean? One of those spots to me is like, I think it'd be.
All right. The 3 rules that are used to name muscles are typically, oops, hold on. Yep. The 3 rules that are typically used to name muscles are the location of the muscle. So as we mentioned, the bicep brachia is in the brachial region of the body. That makes that pretty simple. The second general rule is the action of the muscle. So guess what an adductor does?
Adducts.
It adducts. Guess what a flexor does?
Flexes.
Flexors, there you go. That makes it pretty simple. The third general rule of how something is named is the shape of the muscle. All right? And all of these things, the locations, obviously the action, and the shape of a muscle all influence how they affect the body, what kind of actions they're going to have. So by understanding the shapes of muscles, it can help us to understand the actions that those muscles are going to allow. All right? Questions on that? All right, excellent. Let us then clear all of that and look at these shapes. There are 4 general shapes for a muscle, and those shapes are basically determined by the orientation of the fascicles. Now, we have learned about the anatomy of a muscle. It has muscle fibers, long, thin, thread-like structures that are bundled together into, hold on, I should probably change color of that. Bundled together into a fascicle. Now, when we bundle muscle cells into a fascicle, they are surrounded by connective tissues, and they're all going to be parallel to each other. If you think about it, this is useful. It makes sense. If I contracted muscle cell A, or muscle cell B, or muscle cell C, are they going to have the same effect on the fascicle, or are they going to have different effects on the fascicle? Same, exactly. All of them are going to make the fascicle shorter, and so all of it is going to move in this direction. Doesn't matter whether I use A, doesn't matter whether I use B, doesn't matter whether I use C, doesn't matter if I use A and B together, or B and C together, or all 3 of them together. It's going to be the exact same action. The only difference will be how much force I'm able to generate. So inside of a fascicle, all the muscle cells are parallel. However, if this is fascicle number 1, and I have a different fascicle, fascicle 2, that is oriented in this direction, and of course the muscle cells inside of it are going to all be parallel as well. But if I contract fascicle 1 and I contract fascicle 2, does my muscle move the same for both of those? No. If I contract fascicle 1, my muscle is going to move this way. If I contract fascicle 2, it's going to move not just to the right, but also up. So it'll go up to the right, whereas this one just goes to the right. So it is the arrangement of the fascicles that determine the shape of the muscle and what the muscle can do. So let's write that down. The arrangement of the fascicles determine the shape of the muscle and what the muscle can do. Oops. There. And then I should be able to draw that out. Perfect. All right. We comfortable with that idea?
Yes.
Perfect. So with that in our pocket, the fascicle organization determines the muscle shape and what the muscle can do. Let's look at our 4 major shapes. Here we see on our illustration the 4 major shapes, and let's talk about all 4 of them. The first arrangement of the fascicles is in a parallel muscle. Guess the arrangement of the fascicles in a parallel muscle.
Parallel.
There you go, exactly. Fascicles are parallel. Oops. Parallel, perfect. Excellent. This is also the most common type of muscle arrangement, where all the fascicles are pointing in the same direction, and it forms a fairly functional muscle, right? All the fascicles are pulling all in the same direction, so it is fairly efficient. Notice it gives the muscle kind of that spindle shape with an enlarged central region and then 2 tapering ends where it has those attachments, those tendons to the origin and the insertion. And when this muscle contracts, so notice as this muscle contracts, as we've talked about, it is going to get shorter. Is anything else going to happen to this muscle when it gets shorter? As the 2 ends come together, what happens to the belly?
Gets wider.
Yeah, the belly gets wider. It's going to bulge in the middle. Absolutely. What is the #1 most important reason why every single person, men and women, contract their muscles? Why? What is the #1 reason why everybody contracts their muscles? The most common reason why people contract their muscles? All right, you guys don't seem to be able to come up with it, so I will tell you. It is to woo women. The #1 reason to contract muscles is to woo women, right? There you are on the beach. Some woman comes up to ask you where the restrooms are. And so what do? You do those curls for the girls just so that you can go, oh, it's right over there, right? Flexing that muscle that you can show off that big bulge that occurs when you flex that. And again, whether you're a boy or a girl, it doesn't matter what your flavor is. You flex muscles to woo women. Okay, maybe not.
Speaking from experience, that doesn't work.
I didn't say it was successful. I just said that's why we do it. But we do those curls for the girls just to be able to get those big bulgy muscles. And those are the parallel muscles that we're talking about that get that bulge in the center when the muscle contracts. All right. So again, it has that tapered shape with the tendons at the end. And like we said, we get that bulging of the muscle so that we get that as a result. All right. All right. Excellent. Let's talk about our second. Now, again, remember, because of this one, the advantage of this is since all the fascicles point in the same direction, like I said, it's efficient because they're all going to pull, but you get that bulging muscle, and it's only going to pull in one direction, which is fine for your bicep brachii because your elbow only moves in one direction. However, there are joints where we want more flexibility. And for that, we have a convergent muscle. Notice this convergent muscle has kind of a fan-like appearance, where it has a single attachment point, a small single attachment point, and then from there, the fascicles branch out in a fan-like appearance. This one is going to not give us necessarily quite as much strength as what we got with the other ones because they're not all working together. But what we are going to get with this is a much wider range of motion. Notice if we just flex these superior fascicles, it's going to bring our attachment point up. and towards the left. If we just flex the bottom ones, we're going to be able to bring that attachment point down and to the left. If we just flex the middle points, we are going to bring the muscle straight to the left. One of the areas where you see this, a common example of this, is the pectoralis major, right? You do that pectoralis major, and one of the activities that you do to build this muscle is you do a bench press. However, if your goal is really to work out your pectoralis major, should you do all your bench presses from a bench that is completely flat and horizontal?
No, you should do an incline.
Yeah, sometimes you'll do it inclined, sometimes you'll do it declined. The reason for that is you have more range of motion, and as you're doing those more ranges of motion, you are using all of the fascicles at that different orientation to give you that much broader range of motion that you have with the shoulder than you do with just the elbow. So it gives the advantage of this convergent muscle is it has much more flexibility and motion because of the broader span of those fascicles. All right. Let's look at our third type. The third type is what we call a pennate muscle. Basically, what we have here is two things going on. The first, we have many parallel fascicles. But notice they're parallel to each other, but not to the tendon. Oops. Let's simplify my drawing a little bit. Here in purple is our tendon. And then what we have with our fascicles is a bunch of fascicles that are all parallel to each other, but not parallel to the tendon. Notice it gives it kind of a quill-like appearance, which is where the term pennate comes from. So we have this flaring quill-like appearance with many fascicles attaching to a single tendon. The advantage of this is strength relative to size. Remember when we were talking about playing tug of war, we were talking about how you guys would win over me because all 20 of you were able to get all of your hands, 40 hands onto that rope and pull on your side. So notice the more fascicles we can get on this tendon, the more we're going to be able to pull. Notice if we line these up parallel with the tendon, making that parallel muscle, we have this big fat muscle in that area, right? So if you think about in a small compartment, like, oh, I don't know, your forearm, unless you're Popeye, do you have big, huge, massive muscles in your forearm area? Typically not, but that doesn't mean that you don't have a tremendous amount of grip and wrist strength. That grip and wrist strength comes from these much smaller pennate muscles because they're able to be smaller in size, but still get a massive number of fascicles onto that single tendon. So what we get is a muscle that is relatively strong with a relatively small size. Now, as it turns out, there are three different types, or let's say it this way, three specific types of pennate muscles. The first one, like the one I've drawn here, is what we call a unipennate muscle, where you have one tendon and one row of fascicles. The second is what we call bipennate. Bipennate is where you have one tendon and two rows of fascicles. In fact, one of the strongest muscles in your body is one of your leg muscles called the rectus femoris. The rectus femoris has this long, straight tendon right down the center of it and two big rows of fascicles attaching to it. Because you think about it, biggest chunk of meat you have to move in your body is your leg, right? If you want to lose a quick 30 pounds, cut off a leg, right? And then boom, your weight goes down dramatically as a result of that. It's a big, huge chunk of meat that you have to move around. And so having a big, strong muscle that can help to lift it and swing it around is super important. And so that rectus femoris is that bipennate muscle that has a tremendous amount of strength. Of course, we're not limited to just one tendon. We can actually have multiple tendons, and on those multiple tendons, we can have multiple rows of fascicles to them. And this would give us a lot of strength with a tremendous range of motion. And probably the classic example of this is your deltoid muscle. Your deltoid muscle has multiple tendons with lots of fascicles, giving you tremendous strength to bring the arm up to the side with a relatively small muscle. Now I've drawn these really simplistically. Let's look at the pretty pictures from your textbook that show this. So again, contains a large number of fascicles, producing a lot of tension, but in a relatively small amount of space. Here is our unipennate. Here is that bipennate. Again, we see that single tendon down the center, and then two different rows of fascicles connected to it. And then lastly, oops, here is our bipennate, 3 different tendons with 3 different rows of fascicles attaching to them, giving us tremendous strength in a relatively small amount of space. All right. Excellent. All right, the last and fourth is a circular muscle. In a circular muscle, guess how the fascicles are arranged?
In a circle?
In a circle, there you go. See how convenient that is? Absolutely. So fascicles form a concentric circle. These types of circular muscles are typically found around openings, right? So we've got some around our eyes, some around our mouth that we will talk about in their relationship. And with a circular muscle, when you contract a circular muscle, what happens to the lumen of that circle?
Decreases?
Yeah, it gets smaller, absolutely. So when we contract that, it gets smaller. Now, while we have them on our openings, there are other places in our body where we actually thicken that circular muscle, so much so that when it contracts, not only does the lumen get smaller, but the lumen can actually close. When that occurs, it basically forms a valve-like structure. And what would we call that valve-like structure?
Levatoriana?
Well, so true, but is there a generic term for that, for a valve-like structure other than a valve, a structure? Sphincter, there you go. We would call it a sphincter. There you go. So again, it gets smaller, and some of these can actually thicken to the point where they actually form sphincters, where they actually act as valves, controlling the movement of materials into and out of our body. All right? Questions on that? All right, excellent. With that, we are ready to talk about the muscles. Again, here are the illustrations from your textbook. As you can see, this is just a fraction of the muscles, most of them being superficial muscles that we see from the body. So again, I know that list I've given you is somewhat intimidating when we look at it, but I want to emphasize and remind you that we are looking at the smallest of fractions of the muscles that you could be responsible for in this class, but we're going to be talking about some important ones. All right. We've already done this once because I did this when I did our example, but let's go ahead and talk about the masseter. Here is the masseter. It is a superficial muscle basically over the jaw. As I mentioned, one of the important things for us to be able to do is to actually draw these origins and these insertions. So let's switch to this pretty picture. Excellent. And then, hold on, I want to change the view, get the width. There we go. Excellent. So we can make it a little bit bigger. And as I mentioned, we are going to use red and blue to draw these. So let's start first with the origin. What did we say the origin? And again, remember, you should have your handout in front of you. So these should be super simple, easy questions to answer. What is the origin of the masseter?
Zygomatic arch.
There you go. It is the zygomatic arch. So what you're going to do is you are going to take your pen like I am, and you are going to draw red on your zygomatic arch. Excellent. Might be more convenient if I make that a little bigger. There you go. All right, so there you go. We have our zygomatic arch as our origin. What is our insertion of the masseter?
Mandibular angle.
Mandibular angle.
Perfect. That mandibular angle, remember, was that angle of the jaw that we talked about. Now, remember, as we mentioned, the goal of a muscle is to pull the origin towards the insertion. So what we can do is we can see what joint this crosses. This crosses the jaw joint. And when this muscle contracts, basically it is going to bring the mandible straight up. Of course, we're not going to use a term like straight up. We're going to use appropriate anatomical terms always. So when we do this, we can do it one of two ways, right? Remember, we can identify the part of the body that moves, or we can identify the joint. So to bring up, what is that action again?
Elevate.
Elevate. So we can say the masseter elevates the mandible, or we can say that the masseter elevates the jaw. Both of those are acceptable. It elevates the jaw, it elevates the mandible. And again, we can see here clearly, it can only do one thing. All it can do is bring it up. All right? And what was the example of the activity that I mentioned we would want to use for this?
Take a bite out of an apple.
Yeah, taking a bite, right? That closing of the jaw, bringing it up, right? That is what the masseter does. So that's our goal. We've seen the muscle. We've identified its origin and insertion. We have identified its action. And now on top of that, we have come up with an activity. And again, when it only has one action, maybe we don't need an activity, but we're going to go through the hoops of it anyway to make some sense of it. All right? Do we get how the game's going to be played?
Yep.
Excellent. All right. So let's go back then and switch to a new muscle we haven't talked about yet. Although I will do one thing actually before we do this. Notice if you put, because again, as you guys have mentioned, obviously elevating the mandible is something we do when we chew. So notice if you put your hands on your cheek and you clench your teeth, as you clench your teeth, you can actually feel that masseter muscle contracting. So you can actually, because it's a parallel muscle, it's bulging a little bit. So as we chew, as you clench your teeth, you can actually feel that muscle in your cheek increasing its tension, bulging as size as you clench your teeth while you do that. All right?
Question.
Yes.
I was having this debate with someone earlier. Is the masseter the strongest muscle in the body, or is it the quadriceps?
Well, I would be careful. The quadricep is a muscle group. So those are four muscles working together. What I would say is that the rectus femoris, which is one of the muscles of your leg, is probably one of the strongest per size. So when you think of the amount of strength versus the size of it, I would say that that is a pretty strong muscle. The masseter isn't that strong, right? Again, because it's not the only one that's involved with elevating the mandible. And in fact, that's where we're going next. I would say this muscle we're talking about next is probably stronger than the masseter. And if nothing else, if you just counted the number of fascicles in the temporalis versus the number of fascicles in the masseter, right there might tell you that this might be a slightly stronger muscle. Right? But notice they are similar, not identical in their actions, and they're similar but not identical in their locations. So let's talk about the temporalis. Here is the temporalis. It is a superficial muscle here located on the lateral aspect of the head. If we look at our picture here and we want to draw the origin, what is the origin of the temporalis?
Temporal fossa.
Right. Remember, there's all this hunk and junk at the bottom part of the temporal bone. But up here, basically where the squamosal suture is, all of this part is flat. So all of this part up here is basically just the flat fossa of the temporal bone. And that's where this muscle is found. It is a nice superficial muscle found here on the temporal fossa of the temporal bone. And what is its insertion?
Coronoid process of the mandible.
Coronoid process of the mandible. Remember, the coronoid process is this blade-like portion of the mandible. Now notice it's hidden a little bit by the zygomatic arch. So again, you're doing these drawings to help you to remember it. So if just drawing it like I did it right there helps you to remember that that's the coronoid process. If you want to do it as a dotted line, or I could do it as a highlighter. Oh, that's horrible. To highlight that it is hidden kind of behind the zygomatic. Whatever it does that helps you to recognize that, in fact, if you wanted to form a black line and actually draw it in, even though it's not there to remember you, whatever it is, there's no wrong answer. This is your study guide to help you to be successful. Okay?
Good question.
Yes.
For the test, we won't be required to draw it. We'll just be an arrow pointed to it.
Exactly. So on the lab exam, I will have a mandible there. Now here we're doing drawing, so that's why we're drawing it. But you're absolutely right. I can use one of those pictures of the skull that we had on the past test, and I can point at a mandible, and I can point to the coronoid process of the mandible, and I can say, identify the muscle that inserts into this bone feature. So yes, absolutely. In fact, like I said, probably 20%. Again, so here in the classroom, there are 30 stations for this exam. And of those 30 stations, usually somewhere between 6 and 8 of them have bones sitting at them. So if you think about that, 30 stations, 6 to 8 exams, it's about 20%, something along those lines. So somewhere around close to 20% of this exam is going to be pictures of bones where you're going to have to identify muscles by their origins and their insertions, by these bone features that you're supposed to already know. So that's the other reason we take the time to draw these, because these are things that should be familiar, and if aren't, we want to make sure they are familiar, because I absolutely guarantee you that there will be bone pictures on this lab exam. In fact, let me think. If we think about the lab exam, obviously the majority of the exam is going to be either models or pictures of the arms, legs, torso, etc. Right? Muscles that you're going to be. That is going to be the majority of the exam. There could be some charts of the muscles, but again, it's still going to be pictures of muscles. So it'd be models, pictures. I guess under pictures, I would put charts. So that's definitely going to be there. There's definitely going to be some histology, right? Notice that histology can be both slides, like we've looked at, electron microscopy slides, light microscopy slides. But remember, we also have those histomodels that we've been looking at. So those will definitely be things, that sarcomere model, the two skeletal muscle models are definitely things that will be on the slide. So those will be there. There's definitely going to be bones on it. And again, I will tag the attachment points. And you need to be able to identify the muscle by the origin and the insertion. And as I also mentioned, you will have somewhere between 2 and 3 bodybuilder pictures where you're going to need to recognize muscles from the superficial anatomy of a bodybuilder. And that's something we'll be going through also as we work on this together. Bones, bodybuilder, histology, models, pictures. I think that's it. I can't think of anything else. I'm working my way around the classroom trying to think of all the models and stuff. I think that's it. That's it. Those are the things that are going to be on the exam. So yeah, so absolutely. Bones are definitely going to be one of the things that are on the exam. 2 or 3 pictures of bodybuilder pictures because they're fun. Definitely some histology, but most of it's going to be pictures like the previous ones we've been looking at, or pictures of the models, the things that you get from the practice anatomy lab and those kind of things as well. So that's what the majority of the exam is going to be. Arms, legs, torsos, heads. And you're going to need to recognize the muscle. And then when you recognize the muscle, know its action. And again, that's one of the things that's going to help you on this exam. For instance, if you get confused by the masseter and the temporalis, and I have an arrow pointing at one of the 2 of those, and I ask you to identify the 2 actions of that muscle, which one is it, the masseter or the temporalis?
Temporalis.
Yeah, notice I didn't even have to point at a muscle, because how many actions does the masseter have?
1.
One.
So if it has 1 action and I'm asking for 2, am I pointing at the masseter?
No.
So that's going to be something that helps. However, remember, notice here in our temporalis, muscles pull origins towards insertions. So notice in this case, when this muscle contracts, not only does it bring the mandible up, but it also brings the mandible back. Of course, we're not going to get away with terms like up and back. So what does it mean to go up? What are the 2 actions of this muscle?
Elevate and retract.
Elevate and retract. Excellent. All right. Questions on that? All right, excellent. So, group 2. What is your activity that is going to help us to remember the temporalis? Do I have to actually figure out who group 2 is?
No, it's chewing.
Okay, excellent. Are you going to demonstrate it for us?
Yeah. So just open and close your mouth.
There you go. Bring it back. Absolutely. Yeah. Again, I like to exaggerate these things. So, and again, I'm an older sibling. I have 3 younger sisters. And of course, their names are Ima, Yura, and Iza. No, I do have 3 younger sisters. Those aren't their names, though. It would be pretty bad. But being an older brother, I am required by law to make fun of my younger siblings. And so anytime any of my 3 siblings did anything stupid, which was most of the time, what do? You give them that "you're a big dork" face. And how do you make that big dork face? You elevate and retract your mandible, right? That grrr, you're a big dork, right? That's that elevation and retraction of the mandible. So that, again, it's the same kind of thing. Yes, obviously this is used for chewing, because again, if you put your hands on your temporal and your temples and clench your teeth, you can feel that muscle contracting in there. And in fact, if you put it on your side, you can feel both the masseter and the temporalis at the same time.
Yeah.
All right. Excellent. Nice job, Ariel. Any
Thanks.
question on that one? All right, perfect. Let's move on to the next one. As I've mentioned many times, there is always an alphabet soup term that you are going to be responsible for on this exam, right? Sternocleidomastoid is definitely one of those. You've got to be guaranteed at some point or another you're going to have to spell sternocleidomastoid. However, like most of our alphabet soup terms, it tells you everything about it. The sternocleidomastoid has 3 attachment points. Guess what the 3 attachment points are? Well, sterno refers to what?
Sternum.
Sternum. Clido refers to what?
The sternal end of the clavicle.
Clavicle, excellent. And mastoid refers to what?
Mastoid.
Mastoid process, there you go, exactly. So while it's an alphabet soup term, its name tells you everything about it. Let's take a look at its attachment points. Now, sternocleidomastoid, it has 3 attachment points. It doesn't have 2 true bellies, but it is not a continuous attachment point. So the origin, there are 2 unique origins. One is from the manubrium, the top of the sternum. Then with a little bit of a gap over here on the clavicle, on the sternal end of the clavicle, is the second. So it has 2 different origins where it comes together, and then it inserts into the mastoid process. Now notice I could cheat, and I could draw my arrow over this way for this one to point out that it is over here. Right, and we could draw it in over there. So that's one way we could do this, because again, the point is for you to do this. But what I do is I just cheat and I just draw my mastoid process there, because I know it's behind my ear hole over there on my side of my head. And so I just draw it that way. So whatever helps you to remember it. If you want to draw it over here is fine. If that's too confusing, because you have two other muscles we're going to be putting over here, right? So definitely don't put your arrow there. But you get the idea. Whatever helps you. Oops, no, not what I wanted to do. I want to do that. I don't even know what that is. Go away. There we go. So I just like to draw it here because that helps me to remember what it is. And again, it's in the name as well. All right. So notice we see this here. I've drawn it. Let's go back to the picture from the textbook. Notice it's a deeper muscle. So we don't see it in this view. But notice we peel away a lot of those pesky muscles. Here we get a good view of the sternocleidomastoid. So here, notice this is the portion that attaches to the sternum. This is the portion that attaches to the clavicle. And then it inserts behind the ear to the mastoid process. And that is the sternocleidomastoid. Now, how many actions does the sternocleidomastoid have?
Two.
Two? Count again. How many does it have?
Three. Okay, 3.
It has 3. Now, one of the things you have to remember is you have 2 sternocleidomastoids, one on each side, one on the left and one on the right. Remember, the job of a muscle is to pull the insertion towards the origin. So notice if I use both of them at the same time, what it's going to do is it is going to bring my mastoid process down towards my clavicle and towards my sternum, and it is going to flex my head. In fact, the sternocleidomastoid is sometimes referred to as the prayer muscle because it is the muscle that bows the head in prayer. However, I don't have to contract both of them at the same time. If I just contract one, what's going to happen is it's going to pull my mastoid process towards the sternum, towards the clavicle. And what will happen as it does that is notice it rotates my head. So as we do that, it rotates my head and my chin moves away from the muscle that is contracting. However, as I also mentioned, I can use other muscles to stabilize my head so that when I contract these muscles, instead of the head coming down, my ribs come up. I can elevate my rib cage. I can elevate my sternum. So there are 3 actions. Flex the head, rotate the head, elevate the sternum. Group 5, what'd you come up with for this one?
We just came up with nodding yes and shaking our head no.
Okay. When would you do something like that? Because again, you gotta, so I do like that, like in the classroom, you want to nod your head yes, and it is going to be able to, if you alternate them, shake your head no. So that is definitely an activity where you can put them all together. You're going to demonstrate it for us for the extra point? You don't have to. Okay.
Oh my gosh, I want to, but my camera isn't working.
Are you the only one in your group that's here?
Tim, can you do it? Sorry.
I can do it.
Thank you. I thought I fixed it. I'm sorry.
Can you guys see me?
Yep, we got you.
Okay, so this is a knock head and a check head, right?
Although you're not really bringing up your rib cage when you do that. So that is the only other thing too. Remember, we can reverse it and do it that way. But we get the idea. And so that was good, definitely. We'll give you guys the points for that. However, remember, as I mentioned, I have some favorites, and this is actually my favorite one. I had this, again, this was like 10 years ago. And again, so you got to think of the state of the world 10 years ago is very different from it was, 3 or 4 years ago. And obviously different now. But I had this quiet, demure, a student who sat in the back of the class, barely talked up during class, barely talked up during lab, very diligent, did her work, was there every day, but just very quiet, very demure. She stood up for this muscle and demonstrated her term, not me. The sternocleidomastoid is used for what she referred to as a bong rip, right? You got your bong, flex your head, elevate your rib cage, rotate your head to the side. So there you go. You use your sternocleidomastoid for a bong hit. So that is the action that I use to remember the sternocleidomastoid, and I fully expect nobody to get the 3 actions of the sternocleidomastoid wrong now that they've learned that. All right? Questions on that? And like I said, this was before it was legal. And this was her. Now it's legal. Everybody can do bong rips. But back then, it was hilarious. All right.
Which group we are?
I'm sorry, say again?
How do we know what number group we are?
What number what?
What number of group?
Oh, the groups were assigned last class. So whatever group I put you in. So hold on. I can read them off. I have to go to the class for that. So also on Canvas, it shows what your group was in when you log on for your homepage, but I can tell you right now. So I'll read them off just so that everybody knows. All right, group 1 is Patricia, Sophia, and Amanpreet. Group 2, Michelle, Ryan, Madison, and Ariel. Group 3, Adina, Candid, and Sandy. Group 4, Cody, Stephanie, and Taylor. Group 5, Edna, Finn, and Nicole. And group 6, Alexa, Ursula, and Jessica. All right. So those were the groups. Again, like I said, when you go to your homepage on Canvas, I set up a discussion board so you guys can work on this. Because remember, all you needed for today was the head stuff, because that's all we're going to do. But on Thursday, we're going to do the trunk muscles. We may get into them, start the arm. Definitely the next week or Friday, we'll get into the arm. We'll work our way in through the arm and the legs. So eventually, you're going to need these for all of them. So like I said, I broke us up into groups last time and gave you some class time, but I've also set up the discussion boards so that you can do it asynchronously. You guys obviously can contact yourselves with small groups. Again, remember, it's free if you're just getting a couple people together on Zoom, so you can actually set up your own Zoom classrooms and do this stuff. Or if it's because of your schedules not being compatible, you need to do it asynchronously. I've got it set up on Canvas where each group has their own discussion board. And again, you should have 2 now. There's one for the skeletal, but remember, we've moved on to, actually, maybe I'll close the skeletal one. Although, no, I want you guys being able to communicate. I'll leave that open. Just make sure you're using the bone discussion groups, the muscle discussion groups for this one, because we're doing the muscle stuff. All right? Excellent. All right, any other questions on that? All right, that was the sternocleidomastoid. And after that, oh, and then one last picture of our sternocleidomastoid. And what's nice about this picture of our sternocleidomastoid is it shows us our first muscle group that we are going to be responsible for. The muscle group we are going to be responsible for is the scalenes. Now, again, as I mentioned, it is made up of 3 muscles. And here you see the 3 muscles: posterior scalene, anterior scalene, middle scalene. But as I also mentioned, do you actually need to know that?
No.
Yeah. Okay, excellent. No. So yeah, exactly. You just need to know them as a muscle group. So you don't have to worry about, I'm not going to point, if I point at any of these and ask you the muscle, you just have to say scalenes. Notice on the surface, if you just look at the bellies of these, you can see how they look similar. The sternocleidomastoid is in the neck, the scalenes is in the neck. And again, remember, you obviously have them on both sides. They've just separated them here so that we could see them. They're similar in where the bellies are located, but if you look at the origins and the insertions, you can see that they are different, and that is why they're going to have different actions. Notice in particular, the scalenes doesn't attach to the head. It just attaches to the vertebral column. So let's go back to our drawings and put the scalene in. So the scalenes, we're going to stick on this one over here. What is the origin of the scalenes?
The transverse processes of C2 through C7.
Okay, transverse processes. So these are these things over here of C2, C3, C4. Notice I don't have all of them here, again, this illustration, but we get the idea of C3 through C7. So we might want to write that there just as a reminder. C3 through C7. Oops. As those transverse processes for the origin. And what is the insertion?
The anterior lateral surface of ribs 1 and 2.
Oops, wrong color. Excellent. Rib 1 and rib 2, anterior and lateral. So again, notice the job here, anterior and lateral is the key. When a muscle contracts, it brings the origin, the insertion towards the origin. I hate that that's that big. There we go. Its job is to bring the insertion towards the origin. So obviously one of the effects of this is to elevate the ribs, right? Sternocleidomastoid did the same thing. They both elevate the ribs. They are what we call accessory respiratory muscles, right? As you're sitting here calmly in your chair listening to me lecture, your chest shouldn't be heaving as a result of that, right? Instead, the primary muscle that is doing your normal resting breath is your diaphragm. However, right, if we have to take an exaggerated inhalation, we have that ability to be able to do that because of these accessory muscles like the sternocleidomastoid and the scalenes. However, remember, we can also reverse it around so that we can bring the neck down. And remember, the insertion is anterior and lateral. So if we bring the head down anteriorly, if we use both scalenes together, it is also going to flex the head. So notice flexing the head and elevating the rib cage is something the sternocleidomastoid and the scalenes have in common. But the sternocleidomastoid attaches to the head so it can rotate the head. The scalene attaches to the lateral aspect of the vertebrae. So when we just contract one, it brings the head laterally. And that lateral movement of the head is that lateral flexion. So it flexes the head, laterally flexes the head, and elevates the ribs. All right? Questions on that? Group 1, what do you have for us for this one?
So for the elevating ribs and, oh, hang on, let me turn on my video. Can you see me?
Yep.
Okay, so for the elevating ribs and the flexing the head, it's like you're like crying in public. You have to like hide your head, but then you're also like breathing heavy.
Oh, I like that. I like that very much, right? You're going to go down, you're going to turn your head kind of to the side, but you're also heaving. I like that. I like that very much. Very cool.
And then for the lateral, or does that include the lateral?
I would include the lateral. Again, I think it's better if you can put them all together into one activity, but so yeah. The other one I always think of for this, I always think of like, you know, back in ancient times, there were those dogs that would do that. Like they were doing, it was like one of the recording devices or Memorex or something like that, where the dogs were always kind of tilt their head to the music, right? So I was kind of like, oh, I always kind of think of it as that kind of thing too, but yeah. All right, excellent. All right, good job. And you're group 1, right? Is that what I said? I forgot what I said. Perfect, excellent. All righty, there we go. So we have that. We are rapidly approaching the end, and notice, just this quickly, back to here. So there's the scalenes. We have our second muscle group. Our second muscle group is the zygomaticus. As I mentioned, it is comprised of 2 muscles, the zygomaticus minor and the zygomaticus major, but we don't worry about the individual muscles. We're just going to worry about them independently, collectively as the zygomaticus muscle group. What is the origin, oops, wrong button. What is the origin of the zygomaticus?
Zygomatic bone.
Excellent. Its origin is the zygomatic bone. Notice not the arch, the bone. So that is out here, more the apple of our cheek, right? Not over here on the arch, but more the apple of our cheek. Excellent. And what is its insertion?
The skin and muscles at the corner of the mouth.
There you go. Remember, as we've talked about, most skeletal muscle attaches to bones, but most isn't all. And this is the example. This one actually inserts into the skin and the muscle on the lateral aspect of the mouth. So again, as we think of it, if we are going to contract a muscle, we bring the insertion, oh my goodness. we bring the insertion towards the origin. So as we bring the insertion towards the origin, in this case, we are going to elevate the lateral aspect of the mouth. And group 3, when would you do something like that?
You do that usually for smiling.
Yeah, exactly. You going to demonstrate for us?
Candid very happily took the responsibility.
Okay. Candid, are you still there?
Yeah, I'm here. Sorry, I'm a little bit sick right now, but
It's okay. I can smile.
do you mind, Adina, if you can do it?
It's okay.
Thank you.
Yeah, let me move it. Okay. You guys see me?
Yep.
Okay. So this is the motion that's gonna.
There you go. Perfect. Absolutely. That's perfect. Spectacular. Thank you so much. Excellent. All right. So like I said, some of them are pretty straightforward. So that, again, when they have just one action, it's not as tricky to come up with something. And again, this is an obvious one. And hopefully you guys do lots of smiling during the course of the day. Give that zygomaticus a good workout. All righty. Questions on that? Well, notice just that simply, we have now identified all the muscles that you are responsible for the origins and the insertions of on the head and neck. That doesn't mean we've done all the muscles. We do still have some more muscles we have to talk about. We have to do their actions, and we have to do the activity for them. But there are no more origins and insertions we have to worry about. So let's start first with a slightly deeper muscle. This deeper muscle is the buccinator, and I'm going to cheat and take my highlighter and make it nice and dark to make it easy to find the buccinator. Notice right here is our zygomaticus muscle group. Right here is our masseter. Those are two superficial muscles. And notice right in between them, and I'll use a nice bright color like yellow for this. Right in between them, right here, this muscle right here that is deep, that we can see kind of deep to the side there, that is that buccinator muscle that is deep in there. It is not surprisingly covering the cheek, since the cheek, you remember, is the buccal region of the face. So it's not surprising that one of the major muscles forming your cheek, your buccal region, is the buccinator, right? So that's the muscle. We don't have to worry about the origin and the insertion. So we just have to focus on its action. What is its action? What is the action of the buccinator?
Press the cheek against teeth and lips.
Yeah, so obviously it's going to play a very vital and important role in chewing and speaking, things along those lines. Who had the buccinator? Actually, you all had the buccinator. So group 6, what did you come up with for the buccinator? Did you come up with an important activity or some activity that will help us remember what the buccinator does?
So we came up with like the fish face.
Okay, excellent. You gonna demonstrate it?
How are you? Sure.
Okay.
So it's like you do like that.
There you go.
And you like have your cheeks go in.
The only thing, you're absolutely correct. However, the only thing I would be careful with is you got to be careful of the lips, right? You don't want to puff it too much because that is a different muscle, right? Excellent. And of course, who is the most famous of all of the buccinator users of all time? The greatest movie of all time?
Angela Jolie.
Not a bad guess. That is a good one. No, but even more famous, even more important, even a more transformational movie. How about Blue Steel?
Yep.
There you go. Right? Blue Steel, greatest movie of all time, Zoolander, right? Using that buccinator muscle. He had a well-advanced buccinator muscle, so much so that he was even able to do Magnum, right? There you go. Greatest movie of all time. All right, maybe not, but you get the idea. That Blue Steel, that squeezing of the cheeks, right? That is that buccinator muscle. All right, excellent. Orbicularis oris. Orbicularis meaning circular muscle, oris in the oral area. So this is the round muscle that wraps around the lips. Again, we don't have to worry about origins and insertions, but as we know, When you contract a circular muscle, its lumen gets smaller. So when you contract the orbicularis oris, what does it do to your lips?
Close and protrude.
All right, closes and protrudes them. All right, guess this one leads to group four. Group four, what'd you come up with for the orbicularis oris? Group four?
Sorry. We came up with duck lips.
There you go, exactly, right? Or pursing your lips to whistle, blowing a kiss. You can demonstrate it for us, Cody?
I really don't want to.
Okay, you don't have to. Nobody's required. It's just a
duck
bunch of
lips,
extra credit. So there you go. See, you can take a selfie now. There you go. You're perfect. Excellent. Hey, I did a bong rip. The least you can do is blow a kiss. That can't be that bad. All right. All right. Excellent. There you go. So there's that one. Notice it is not our only circular muscle. We also have the orbicularis oculi. The orbicularis oculi is a circular muscle around the eye. And let's pick on group four again. Cody, what do you got for the orbicular, or anybody for your group? Doesn't have to be you, Cody. What did you guys come up with the orbicularis oculi?
Winking.
Winking. And again, one of the key things to remember about this is a circular muscle. I'm not talking about just lowering the eyelids. Your eyelids are a different muscle, the later palpebrae, which again is not a muscle you're responsible for. So we're not talking about just blinking or closing your eyes. We're talking about a squeezing of the eye, squinting of the eye. So like for that nice big exaggerated wink, or like if you're riding through that dust storm and you close your eyes about that, it's really that squeezing of the eye for that. And did you demonstrate that for us? I missed it. You can tell me that you did if you did. Okay, there you go. Excellent, perfect. Excellent. See what I tell you? It's all used for wooing women. There you go. Exactly. So we have that orbicularis oculi that we do for that. Here is our first multi-bellied muscle. Remember, when we're doing this multi-bellied muscle, one of those magical terms I will ask on the exam is to be specific. So here, this muscle is known as the occipital frontalis. Again, it's a big alphabet soup term, but once again, the name tells you everything about it. There are two bellies for this muscle. Guess where one of the bellies is located?
The frontal bone.
Yeah, on top of the frontal bone. Guess where the other one is located?
Occipital.
Occipital bone. So we have these two bellies. Obviously, the specific one we're looking right now is the frontal belly, but notice if we turn the picture, here we can see the frontal belly, here we can see the occipital belly. Now, I will tell you right now, this muscle is also sometimes referred to as the epicranius, and you are welcome to use that term if you would like. It is an acceptable anatomical term, but you still, on the exam, I'm going to point at one of these two bellies and say, identify this muscle, be specific. So you still need to say this is the frontal belly of the epicranius. This is the occipital belly of the epicranius. So either way is fine. I like occipital frontalis. It's a few more letters, but it tells you everything about it. All right, what is its function?
Elevate eyebrows.
Elevate the eyebrows, absolutely. Right, perfect. Group one, what'd you come up with for this? Group one?
Elevating the eyebrows.
Right, and when would you do something like that?
When you're mad or something?
Sure, or surprised or something along those lines. There you go, exactly. Great demonstration. Beautiful job, excellent, right? Elevating those eyebrows, right? Again, however, it does absolutely elevate the eyebrows, and I'm not sure how well this will show up. All right, I'm gonna completely embarrass myself, so let's stop sharing this for a second. All right, when you have the advantage of not just having a 4-head, but having a 5-head, what you really get an appreciation for is that these two muscles are attached to each other. Because when you, especially if you do it quickly, elevate your eyebrows, you can see that the entire scalp moves as a result of that. So again, these two really are connected to each other. So obviously it elevates the eyebrows, but you can see it really moves your own scalp. You can put your hand on your head and do that, and you can feel the scalp moving back and forth as it does that. The reason for that, as we can see nicely in this picture, oh, I need my annotation stuff there, is that there is a big, large, broad tendon that connects these two bellies together. And of course, as we've talked about, the fancy name we give for a big, broad, flat tendon is an aponeurosis. So you'll notice one more thing on your list. At the very bottom of the page, you will see in bold letters some additional structures you are responsible for. That will be the key word I will use, structure, because these are not muscles. These are just structures associated with the muscles. And so on the exam, I could have an arrow or an X or a star here on this structure and ask you to identify this structure, and you would say that it is the epicranial aponeurosis. All right?
Got any tricks on how to remember that?
I'm sorry?
Got any tricks on how to remember that one?
So again, if you think about it, there's only one structure in the head that you're responsible for. So if I'm asking you for a structure of the head, you know it's going to be this. The other thing too is, again, it's in the name, epicranial. Epi means on top, cranial means on top of the cranium, on top of the skull. And it's a broad, flat tendon. You just have to remember and learn that a broad, flat tendon, and there's going to be 2 or 3 we're going to be responsible for, are known as an aponeurosis. So it's the aponeurosis, it's the broad, flat tendon on top of the head. It's the epicranial aponeurosis. And it's the only structure you're responsible for on the head. So if you see the head and you see me asking you what a structure is, you don't even have to look at the arrow. You know the only structure you're going to be responsible for on the head is the epicranial aponeurosis.
My partner came up with something. It might not, I mean, people might have other better options, but I really liked it. So the analogy was that, let's say someone is preparing a surprise for you.
Yeah.
With your back turned to them, so your occipital bone is turned to them.
Okay.
And they tell you that their surprise is ready, so you show them that you turn around, and now your frontalis is facing them.
There you go.
And then you're surprised because it was a surprise, and then your eyebrows are raised.
Oh, I like that. There you go. So they make you turn around. So first the back of your head's facing them, right? Then the front, and then you're surprised it comes up. I like that. That does definitely put it all together. So that definitely helps for the 2 bellies in the name. All right.
Did you have any favorite way to remember that from past classes?
No, like I said, the ones that I have that I really like, I'll pass on to you. But like I said, for me, it's about the whole fact that my whole head, my whole scalp moves when I move that. That is what helps me to remember that it all connects together. And it's held together by something.
Mm-hmm.
All right. One more, I believe, right? Whoops, I'm doing stars all over.
Platysma.
Platysma. Excellent.
Platysma.
The platysma, and again, I don't care how you say these things as long as you spell them correctly on the exam. The platysma is a big, and again, you're not responsible for the origin and the insertion of this, but it is important to know it is a big, broad, flat muscle. As you can see, it's a superficial, big, broad, flat muscle that covers the neck, but notice it also comes over the chin onto the anterior part of your mandible. Knowing and understanding that helps us to understand its function. Because being under the jaw, what is the obvious action that it will have?
It will depress the mandible.
Right, depress the mandible, bring it down. But because it comes up over the jaw onto the lower lip, it can also affect the lower lip, causing the lower lip to do what?
Pout.
There you go, exactly. Protruding the lower lip, pouting the lower lip, exactly. And so that is what this muscle is able to do. It's able to bring the jaw down and pout that lower lip. Now, notice I didn't put it as one of the actions because it isn't so much an action, it was just putting tension in it. Remember, one of the things that we talked about is that if you used your masseter and temporalis, for instance, to keep your jaw closed, when you contracted the platysma wouldn't bring the jaw down because your other muscles are holding it up. But you would still get tension in it. And so what you get is you get that Hulk neck.
right? You get that hook neck where you can feel the tension in the muscle as that hook neck sticks out.
So that hook neck, when you get angry, is that platysma with that tension in it while you're clenching your jaw. So again, it isn't so much an action, but it's a way that you can really feel what that platysma is, that superficial muscle draped over the neck when you do that angry, gritting your teeth contraction of that platysma muscle. All right? Questions on that? So again, notice if you put all of these together, the muscles that we were just talking about, what I tell you is the most important reason to contract muscles? Why do you contract muscles? Come on. What? There you go. Not just for the girls. See, if you say for the girls, it doesn't sound appropriate. You have to say to woo women, right? If you say to woo women, it doesn't sound so bad. But again, if you think about all the things we just talked about, you use your platysma and you use your orbiculus oris to, you know, whistle, right? When you see that pretty girl, you can raise your eyebrows at her. You give her that big wink, right? You give her that smile using all of those. And of course, when she still walks away, well, you're sad and you're proud as a result of it. So again, it's all involving wooing women. Or again, if it makes you feel better, you can woo One Direction, right? There's Harry Styles walking across the beach. You can do all the same stuff to him. Whatever it is, it's all for wooing. Muscles are for wooing. All right, questions on that? All right, there you go. Just that simply then, we have gone through the muscles. We have really, this is the game. Like I mentioned, for the next 4 lectures, so basically 2 weeks worth of lectures we're going to be doing, this is exactly what we're going to be doing. We are going to go through the physiology of the muscles, talking about how it contracts and continuing to move forward from that. Then we'll come here to lab, doing origins, insertions, axons, making funny faces or swinging our arms around in space, laughing at each other, and then moving on to the next one. So that's the game plan for the next, I keep saying 2 weeks because that's normally what it is, but really the next week, because we have 4 more lectures. We just are cramming it into 1 week's worth of time. Questions on any of that? All right, excellent. That is everything that I got for you today. We get to finish a little bit early because this has been great. We got through everything that I wanted to get through for today. So like I said, continue to work on your groups. We are moving on to the trunk, muscles that move the trunk on, oh, great question. Muscles that move to the trunk on the, on Thursday. So we'll do that and maybe start the arm. I don't know if we'll get far into the arm. There's a lot of trunk muscles. It'll depend on how long the lecture takes. I am still working on the test. Like I said, I have the lab exams graded, but I haven't curved them yet. I need to finish grading the lectures. I'm a little behind on that, but the good news is that is all I have to do today and all I have to do tomorrow. So I'm hopeful to have them completed tomorrow. So my guess is that the grades will be posted sometime tomorrow. And then my plan is for the grades to be completed sometimes tomorrow, and I'll post the grades once I get the curve. And then like before on Thursday, then at the end of class on Thursday, I will release the lectures so that you can take a look at them. You can see my comments. You can see what you got right and what you got wrong. And then again, that'll be for 24 hours, and then I'll close it again on Friday. So that will be the game plan for that. So I anticipate having them done tomorrow, hopefully tomorrow evening, not too late. So that you'll get the grades definitely sometime tomorrow, hopefully, definitely. And then I hope to have them, and then I will release them to you on Thursday. So that's the game plan. All right? Yes.
Just to confirm what you already said. So right now we just have to communicate within our groups of just these trunk muscles, right? Not the upper extremity or anything after that, just the trunk.
Well, so what I would say is you are eventually going to get to the rest of the body. I mean, again, from Monday to Tuesday, you didn't have a lot of time. So obviously I wanted you guys to focus on the head. Eventually, you're going to need to come up with these activities for all the muscles that are on your list. So if you can use today and tomorrow to get ahead, then I think that would definitely be a good thing. But yes, in practical terms, we definitely won't get to the legs this week. So the legs won't be next week. And Thursday, I hope to get through all of the trunk and hope to start the arms. Whether we're able to accomplish that or not, I don't know. But we will definitely get all of the trunk and at least some of the arms by Friday. So yeah, so I would use this time. Oh, speaking of time and tomorrow, I want to also encourage you to spend time, if you haven't been doing so already and have the opportunity, please go to the open lab to speak with Jeff. I know he's got a lot of the models and he's happy to go through them for you. It's nice seeing these pictures. These are nice, pretty pictures, but looking at the models, and most importantly, he can turn and rotate the models for you, is a really great way of seeing how these things relate to each other. So I'd encourage you to take advantage of Jeff in the open lab for learning the material. Yes, you get the carrot of also getting extra credit by doing it, but do it so that you get to learn the information. All right? Great questions. Any others? All right, well then you guys be happy, be safe, enjoy your one day off, study hard. Again, a week from Thursday is our exam. So now's your opportunity tonight and tomorrow to start getting ahead, looking at this material, getting ahead, being prepared so that you're ready, because 1 week from Thursday is our next exam. All right, be happy, be safe, and I'll see you guys on Thursday.